Vasudevan, the co-founder of and Chief Operating Officer of OneCell Diagnostics. I welcome you all this wonderful evening. I would like to call upon Mr. G.R.R. Vittala, who is the Director Strategic Business Development, to welcome all you delegates. Thank you.
Namaste to one and all here. Respected doctors, good evening, good morning, and good afternoon to all the doctors. We welcome one and all to this splendid gathering, and we are grateful for your time and presence on this auspicious day of Gudi Padwa and Chandramana Yugadi, the beginning of the new year and Samvatsar. Due to certain COVID restrictions scenario, we have invited around 50 oncologists to Pune at this venue. I would like to welcome all the doctors who have traveled in person. I would also like to welcome eminent faculty, Dr. Klaus Pantel, Professor Dr. Catherine Alex Penaberis, Dr. Ken Bloom, and Professor Dr. Sinduri A. Maning, Dr. Raja Pramanik, and around 300 number of delegates from 17 countries and 14 cities in India who have joined us virtually for this launch program. I would like to mention and acknowledge all the oncologists and clinicians through this Subhashitani. Aturo cha pita vaidyaha swasti bhuto cha bandavaha gate roge kruti swastye vaidyo bhavati palakaha. The translation goes like this: for a sick person, a doctor is like his father, and while a person is healthy, the doctor is like his friend or a relative. But when one gets cured of his ailment and regains his lost health by the treatment given by the doctor becomes his guardian or a protector. Precision oncology is rapidly evolving in recent years, leading to the discovery of many important markers with diagnostics, prognostics, genomic sequencing, and therapeutic findings. Today, OneCell DX is proud to be uniting many oncologists to launch Precision Oncology 2 for the benefit of Indian cancer patients. Research, education, and quality genomics are the guiding light of OneCell Diagnostics, which is that every patient, everywhere, everyone should receive quality genomic analysis, cancer care that results in the best possible outcome. We are coming together to work in a new ways of identifying challenges and solutions together. The world needs these new tools and it needs them fast. Each of us are doing a great work and request all to join hands for a common goal to help patient lives. Being in oncology field for over 2 decades now, I became increasingly committed to improving outcomes of all people with cancer through you. And that's one reason why I'm very proud to be part of OneCell Diagnostics. This is all from me, your Vittala. Thank you for being a patient listener. We will interact with you all post this launch program. Till then, enjoy your evening. I would now like to invite Mr. Mohan Uttarwar, CEO of OneCell Diagnostics, on stage to give an overview of OneCell Diagnostics. Mr. Mohan Uttarwar, for over past 2 decades of applying innovations in technology to impact human health, including founding companies such as Bioimagine, who pioneered the field of digital pathology, acquired by Roche, and OncoMDx for molecular pathology. Dr. Mohan Uttarwar, please.
Thank you. Thank you, Vittala. You know, serial entrepreneurship sounds like serial killer. Trust me, other than the damn cancer cells, I don't want to kill nobody. Good evening, good morning, good afternoon. Thank you for joining us at a very opportune time. It's an exciting time because all the competencies are coming together to really do something about this deadly disease that we, all are here to conquer. So basically, the whole idea here is that the action over the next decade is going to be at the intersection of science and technology. I realize it's a big topic, and I'm standing in between you and the exciting line of speakers. So I'll answer 5 simple questions in our next 5 minutes. What do we do? Who we are? Why this is important, what we do? Why now? Why us? And if our dreams come true, what's the impact, the societal impact we can make? As we all know, cancer is a complex disease. And complex diseases like that, one shoe doesn't fit all. You need a custom solution, individualized, personalized. And so if you believe that precision oncology is the future, then dreams come true. Multiomics biomarker would be the future of precision oncology, precision medicine. Cancer, as much as it's a disease, it's a data problem. And the complex and the big data needs the tools like AI-enabled analysis. Historically, genomics was the first wave of precision medicine, but now the field has evolved into multiomics. Tissue has always been an issue. So liquid biopsy is the future, but concordance, complexities, etc., are there. But if you really believe that the monitoring is the key aspect, then you don't have a choice but the liquid biopsy. And that's really what we are. We want to be the first-in-class liquid biopsy precision oncology company that brings together both the innovation in science and innovation in technology. Now you ask the question, why? Well, we just heard that every minute there is one casualty. We all know metastasis is the real cause of that casualty. Isn't that avoidable? It is. But you need to monitor. You need to have the tools to monitor proactively, not reactively after something happens. Okay. And I think this is the beauty of the innovation in technology, where we would potentially have the ability to transform that, and the old paradigm of diagnose, treat, and hope for cure will be changed to monitor, screen, predict, and prevent. That predicted the aspect where AI and ML comes in very handy, working in conjunction with this. So we believe multimodal, multiomics, genomics, proteomics, particularly imaging, integrated into this is going to be the future. And we think that future is now. Okay, and that's why the why now part. It's a perfect storm. Action is at that intersection of technology and science. With power and computational, with HPC going on, with AI, ML, and of course, the genomic revolution now turning into the CRISPR, together gives us that opportunity to do something about it. And that leads to why us. Why us is simple. 3 T's as I always look at: the team, the technology, and traction with the key opinion leaders. Very fortunate to have an incredible team, multidisciplinary, 50 of us, and coming from different backgrounds, from material science, life sciences, data sciences, software, also, the medical sciences coming together. We're very fortunate, with over a couple dozen PhDs working together with a common goal, with a common mission, addressing to help this particular stuff. Okay. The technology, very deep IP. IP is the lifeblood of any startup, and here, we do have incredible amount of innovation and also support from key opinion leaders. This whole journey started with Parker Institute of Immunocancer Therapy and 6 of their collaborators, Stanford, UCLA, UCSF, Sloan Kettering, MD Anderson, where they were looking at opportunities to do translation research to speed up, the immuno-oncology trials. And that's how this whole platform was built. I'm very pleased to tell you, we look at it as 5 pillars of innovation, CTCs, and we'll hear more about, this whole thing. 10 times the sensitivity at 1/10 the cost. That's phenomenal transformation. And it's not just that, that's the beginning. CTC is not new, but what we look at as CTC 2.0 with a vision of isolating those handful cells from billions of cells, isolating them, enumerating them, sequencing them, and then applying this multimodal, multi-omics analytics with the AI prediction model and that sort of stuff. Lastly, there's a lot of data. Anybody can do sequencing. So what's the big deal? It's not about generating that data. It's about what do you do with that data? How do you translate that data into actionable insights? This is where secondary, tertiary analysis, integrating the research data with real world evidence data, gives us that ability to have the predictive models, which will be able to help the patients. And that's where the vision of integrated precision oncology console, where we enable and empower practicing oncologists who have incredibly difficult job. In a matter of 5 to 7 minutes, you're making life-changing critical clinical decisions. Okay, this is where our technology platform will come in handy to help them, make anything that they need at their fingertips. We almost think of oncologists like the captain in a cockpit, where if we can provide them, the tools that they need that can navigate them through the data maze, that's the game. Again, you can sing the technology song all day long, but what matters at the end is really service and support. It's not just the high tech, it's the high tech, high touch. And fortunately, we have, a great set of advisors, Dr. Nirmal Rao, Dr. Sujit Joshi, Dr. Vikas Zadav, multidisciplinary team that will bring together to provide, a very high level on-demand precision oncology advice, including Dr. Rala Chougule. So, they provide the platform, set of applications, and service around it that will give you complete end-to-end solution that will help you, get off the fence from a traditional practice to, a precision oncology practice. Our commitment is that of quality, turnaround time, and the cost. And the cost again comes from our fundamental principles, design principle of what we call factor 5 design. Breakthroughs, what takes thousands of dollars because you have to send the samples to the United States, no more. You don't have to spend $4,000, $5,000, wait for 4 or 5 weeks, only to find out, okay, that there's no information there. That's not on. Applying innovation in technology such as, image analytics to really prediction of gene fusions, to the prediction of the outcomes, that is going to the breakthrough. AI is exciting, but it needs the data. If data is the new oil, India is the place for that. And if you put together, this innovation in technology and collaborate with the thought leading, folks here, I think there is absolutely hope to make cancer a chronic disease. And that's our goal. With the innovation in technology, making it affordable, our goal, our mission is to impact the lives of at least 1 million cancer patients. And what I believe firmly is that that time has come where India can play a very vital role. And what I call reverse innovation. If that solution works here, it will work anywhere else in the world. So help us help you make a change, make an impact in the 1 million patients' life. Thank you very much.
Thank you, Mohan, for that inspiring speech. We would now begin with the first session of the evening, which is the unveiling of our liquid biopsy comprehensive genomic panel. I would like to invite the chairpersons for the session, Dr. Kumar Prabhash, Dr. Arun Jamkar, and Dr. Hemant Malhotra will join us online. So please. Thank you. I would now like to invite Dr. Kumar Prabhash to introduce our first keynote speaker, Dr. Klaus Pantel.
So good evening, everyone. And the first lecture is by Professor Dr. Klaus Pantel, and he is a chairman, European Society of Liquid Biopsy. And let's hear him. Thank you.
Well, hello, everybody. It's really my pleasure to be invited here to this excellent meeting today. And I hope that you can hear me.
Yes, sir, we can hear you. Go ahead, sir.
Super. Great. So in the next 20 minutes, I would like to guide you through my ideas and opinion about the past, present, and future of liquid biopsy. These are my disclosure informations. So liquid biopsy has been defined by Katrina Lix-Panabir and me as the analysis of circulating tumor cells in the beginning around 2010. But then later on, it has been adapted also to the circulating products released from tumor cells, particularly the DNA, but also the RNA and the extracellular vesicles. And it has been also now included the host cells into that definition. The key fluid to analyze is the blood. Of course, what is the rationale? Tissue biopsies are the center of cancer diagnostics, but these needle biopsies are invasive and some locations are difficult to access. A single biopsy of a metastatic lesion can miss relevant tumor clones because different metastatic lesions may have different genomic composition. So there is a marked intra-patient tumor heterogeneity. The blood is a pool of tumor cells and their products released from primary or metastatic lesions, including all locations, so it can give some more comprehensive information. And also we can take blood samples sequentially and monitor actually tumor evolution in the context of cancer therapies. And this, of course, gives completely new insights in the development of resistance. So in conclusion, the vision is to get a more comprehensive and real-time tumor information by the analysis of blood or other body fluids. Interestingly, liquid biopsies, there is some background noise now. Somebody, okay, that's good now. Interestingly, in the last edition of the Nature Milestones, Diana Romero has highlighted liquid biopsy as one of the key discoveries of the last 20 years. In Europe, we had a consortium called Cancer ID, which was actually meant to close the gap between the wonderful discoveries published in the academic papers, but also the lack of new biomarkers in the clinic. So this consortium was a private-public partnership of 40 partners from academia and industry. It was funded 5 years with 80 million by the European Union and the participating companies, and it was a very successful collaboration here. At the end of 2019, we decided to sustain that network, and we established the European Liquid Biopsy Society, and I'm the current chairman. And we have more than 60 institutions now from academia and industry. And we also invite everybody from not only Europe, but from all over the world. If you want to join our consortium, please write me a mail. What are the goals? We want to foster the introduction of liquid biopsy to clinical practice, really make it become possible for cancer patients to profit from these research discoveries, encourage the interaction between academia and industry, provide a partner for regulatory agencies, healthcare providers, and patient advocacy groups, support the implementation of liquid biopsy tests into the clinical trials, develop guidelines and provide training, and disseminate the knowledge about liquid biopsy. We have also made a link to the US by being the founding member of the International Liquid Biopsy Standardization Alliance, which is coordinated by the foundation of the NIH. And we wrote a paper together with all our colleagues from the US. So liquid biopsy allows a comprehensive assessment of circulating blood biomarkers. Where are these cells come from? In early stage cancer, the majority of circulating tumor cells are of course derived from the primary tumor. In later stages, lymph node metastases contribute. And in even later stages, when you have metastatic disease, the metastases, for example, in the bone, the liver, or the brain, can contribute and release cells and cellular products into the blood. So it depends always on the stage of the disease, where the tumor cells originate from. The blood is the main fluid for analysis, but of course it can be also, the technologies can be also used to analyze the cerebrospinal fluids, sputum, bone marrow aspirates, urine, or cyst fluids. In the blood, you have two fractions, the cellular fraction, which contains the circulating tumor cells or CTCs as single cells or as clusters, but also circulating host cells such as immune cells, endothelial cells, or cancer-associated fibroblasts. In the plasma fraction, you have the extracellular vesicles, the proteins, the RNAs, the tumor-derived DNA, and also the tumor-educated platelets. So you have a whole variety of analytes that you can analyze from a single blood tube. The technical challenge, and I don't go into any details today, is really the low concentration of CTCs and ctDNA, but there have been excellent technologies developed to overcome this challenge. The two key analytes that are used in liquid biopsy today are the CTCs and the ctDNA. And for the CTCs, we have 3 steps of technology assays. We first do an enrichment of the CTCs based on the biological or physical properties that distinguish the CTCs from the blood cells. Then we do a single cell detection using immunocytology or PCR or the EpiSpot assay. And then we can do a downstream characterization of these single cells at the DNA, RNA, or protein level. For the ctDNA, of course, we are restricted to the analysis of DNA, and there are very elegant methods that can really look at the different aberrations that are known to be present in cancer. More importantly is also that we need always a harmonization of methods and the technical validation, and we did this in our European Liquid Biopsy Consortium and Cancer ID Consortium. And we published the results of this technical validation. We performed large-scale ring experiments. And if you're interested how the different technologies performed, I can refer you to these publications that we recently submitted, and it was accepted in clinical chemistry. So for the CTCs, they have a low concentration of 1 cell per million blood cell. But also the ctDNA is actually a low concentration because most of the circulating DNA in the blood is derived from normal cells. And the tumor cell fraction that we call ctDNA, T for tumor, also is a very low one. So in metastatic patients, you may have 1% ctDNA or more, but in non-metastatic earlier stage patients, you have far below 1% tumor DNA. So you will need really very sensitive methods too. And you have to be careful because if you use these very sensitive methods, your cancer mutations may be derived from normal leukocytes. And so you have to be very careful that you really detect the tumor signal. While when you analyze circulating tumor cells, you're sure you have a tumor cell and whatever you analyze in there is tumor derived. So what are the clinical applications? We have first early detection or diagnosis of cancer, which of course is a very important application. Everybody wants to detect tumors at an early stage to treat them, for example, by surgery and cure the patients. The second one is to follow up the cancer patients and analyze the CTCs and DNA for the real-time monitoring of the response to the adjuvant therapies. The third one is analyze the tumor evolution also in advanced stages and look for the emergence of molecular alterations associated with resistance to cancer therapy. And the last one is detection of minimal residual disease, which means the presence of tumor cells in the body in a patient that is supposedly cured, but there are really tumor cells left that are not detected by any current imaging procedures. So just to go through these different applications, I think for early detection, we have in Hamburg started the Hamburg City Health Study, which actually means that 45,000 individuals, citizens of Hamburg, between age 45 and 74 come to Hamburg. We collect a lot of biomaterial, including 120 ml of plasma, blood plasma, and we get a lot of data, and we follow up also the cancer patients. We have in Europe 2 early detection programs funded by the European Union. One is the European Liquid Biopsy Academy, ELBA, which is focused on the detection of lung cancer. And the second one is called Prolipsy. It's focused on the detection of prostate cancer. We have also lately used circulating proteins, and for example, a circulating stress protein called CYR61, to see whether we can use also these new proteins in addition to the CTCs and DNA for early detection. And this is just one publication that came out lately in Clinical Chemistry. We have really shown that this protein is increased in carcinoma patients and already in early disease in DCIS patients compared to healthy controls, but also non-cancer disease controls. And at 100% specificity, it could detect more than 70% of the early stage cancers. And I think it could be added to some other panels because it seems to be a good candidate. The second application is, can we use CTCs to actually do a better tumor staging? We have the tumor staging, of course, in the clinic, but we know it's not perfect. And CTC counts at initial cancer diagnosis are associated with an unfavorable prognosis. We have shown that in different tumor types and different solid tumor types. And I think that also the One Cell Diagnostics team, has lately published a very nice publication on the prognostic relevance of CTCs in head and neck cancer. So we can actually use the CTCs as an enrichment tool, a tool to study a high-risk population of cancer patients which may need more aggressive therapy. And this, I think, has been already included in the staging manual for breast cancer as a new category called CM0 brackets I+, which means no presence of overt metastasis, but signals for isolated tumor cells. The second application is really to detect minimal residual disease. So after the initial detection of cancer, we apply surgery, radiotherapy, or chemotherapy, or other adjuvant therapies, and we decrease the tumor burden. But quite a few patients have a minimal residual disease left, so there are tumor cells left that are not seen by imaging, but we can now detect those by sequential blood analysis. And this gives us now the chance to do an additional type of clinical trials, not just in the adjuvant setting or in the advanced setting, but also in between. So can we detect actually in the blood a signal for MRD? The answer is yes. This study we carried out in breast cancer patients where we took blood samples years after adjuvant chemotherapy, and the CTCs that we detected there were derived from some occult micrometastasis because the circulation time is very low and the primary tumor was already taken out years ago. And this hypothesis is also supported by the fact that if we find these CTCs 2 years later, which is reflected here by the red curve, that these patients have a worse overall and disease-free survival. We can also, of course, use ctDNA for the same purpose. This is one paper from the group from London, Charles Coombs and Shaw. And they could also show that if they detect ctDNA during the follow-up period here, the red curves, the prognosis is poor. And they could also show that they can detect this signal in blood with a lead time of more than 600 times before imaging can detect such a relapse. So how can we use liquid biopsy in the advanced metastatic patients? Well, we can monitor the circulating tumor cells and DNA counts, and we can characterize them to get more information on therapeutic targets and strategies. Here, together with Katrina Lix-Panabia, we just reviewed in the 10th anniversary issue of Cancer Discovery the different drugs that actually require a genomic analysis, or the drugs that require a transcript and protein analysis. And of course, the genomic analysis you can perform on the ctDNA or the CTCs, but transcript and protein analysis you can only perform on the CTCs, not on the DNA, of course. And that's very important to know. So I give you a few examples. Here in breast cancer, we developed a staining method to type the immunocytochemical expression of the HER2 oncogene. And we could see actually that there is a discordance between the original primary tumor HER2 status and the CTC status at the time of metastatic relapse. So we started many years ago a randomized trial where we treated patients with HER2-negative primary tumors or HER2-positive CTCs at the time of relapse with an anti-HER2 therapy, lapatinib. And this Kaplan-Meier overall survival curve shows you here that the patients who received the lapatinib anti-HER2 therapy, in addition to the standard therapy, the red curve, had a better prognosis than the patients that had received the standard therapy. So we could use this actually to predict outcome and steer therapy. Of course, in oncology, immunocheckpoint inhibition therapy has really opened a completely new avenue for cancer treatment. And we have actually done now, and we are currently doing now, monitoring studies of DNA and CTCs in non-small cell lung cancer and in melanoma, which are both responsible to this type of therapy, but only a fraction of therapy responds. So we need to know which is this fraction and why do the other patients not respond. So we combine now immune markers on the CTCs, for example, class 1 molecules or PD-L1 expression, which is one of these checkpoint inhibitors, and combine that with the information on the DNA. And we do that also now in an international network on cancer immunotherapy, which has been recently established by the Nobel Prize winner Tasuku Honru from Kyoto. In melanoma, we also can detect circulating tumor cells using tumor-specific markers, and we also use exclusion markers so that we don't get false positive results. So we exclude the staining of leukocytes and endothelial cells. Then we can pick the cells manually. We can also do a whole genome amplification, so we have enough DNA for analysis, and then we can do mutation detection. For example, for BRAF mutations, which are relevant to therapy in melanoma. We can also do mutation detection without NGS on the ctDNA. We have developed here a new method, which is cheaper and more rapid, where you can look at 65 mutations of 13 genes, including, for example, BRAF, NRAS, and so on. And I think it's a method that has a quite nice sensitivity of around 0.1% mutation frequency. One other question is if there is something circulating in the blood, if you have circulating tumor cells in the blood, where are they derived from? What is the origin of these cells? And I think this is important to know where you should actually target your imaging. And one challenge are brain metastases because of the blood-brain barrier. So how can we improve the sensitivity of CTC detection in patients with brain metastases? We have done that here actually by introducing new antibodies to capture actually these CTCs. So in addition to the usual EpCAM antibodies, we also added antibodies against HER3 and EGFR, and that could actually lead to a better capturing of CTCs in patients with brain metastases. And once we have detected these cells, we can also again pick these cells, then we can look at mutations, and then we can compare the primary tumor and the CTCs to actually validate our method. And I think this also shows you just one example that also from the analysis of circulating tumor cells in patients with brain metastases, you can obtain genomic information. We have also included this assay in the context of radiotherapy of brain metastases. So we monitored actually the blood during the radiotherapy before and after, and at the control MRI. And as you can see, there are really various curves that the number of circulating tumor cells can change over time during the irradiation. There might be even an increase post-radiation that then drops again. And we have correlated that also to the outcome. Of course, in order to know where the CTCs are derived from, we can perform genomic analysis. We did here an exome-based sequencing of single CTCs in breast cancer patients with brain metastasis. And you can see that many of these cells had the same gains, which are indicated in green here, or they had the same losses indicated in red. So there was a high clonality of these CTCs, and they had specific aberrations that were different from other locations and may help us to conclude that there might be a brain metastasis. We have also analyzed brain metastasis-derived CTCs on the expressional level here for the expression of ALCAM. And we could also show that the expression on the CTCs here in this pilot study on 4 patients was similar to the expression on the brain metastasis. So we may get some information on the brain metastasis by analyzing circulating tumor cells. So at the end, what is the future? I think the future also goes in the direction to do functional analysis. So we have established experimental model to understand the biology of CTCs, find new targeting approaches, and do a drug screening. And one of these cell lines is derived from an ER-positive breast cancer, and we have deeply characterized those cell line, and it was really highlighted at the cover of the EMBO Molecular Medicine. And we have shown that this cell line has similar genomic aberrations in the primary CTC, so it's a good model, and it's also stable in culture, which also means that it's not changing phenotype all the time. The cell line also, after injection into immunodeficient mice, grows at the injection site, and it actually forms metastases that are relevant to breast cancer, and particularly bone metastases, which are frequent in ER-positive breast cancer. So what is the future beyond CTCs and ctDNA? I think that extracellular vesicles, exosomes, and others are becoming very interesting new biomarkers. Exosomes are interesting because they contain proteins, RNA, DNA, lipids, many components of cells. They are released from cancer cells and host cells, and they are taken up by recipient cells, and they can also affect the biology of the recipient cells. So they're also involved in the biological process that actually will dictate the outcome of cancer patients. The other biomarker type are circulating cell-free microRNAs. We have published some reviews on this biomarker. Just in short, I think these biomarkers can have profound regulatory effects. They can be located in exosomes, but also on free-floating proteins. And we have published quite a bit of work how these biomarkers can be used for cancer diagnosis and also monitoring of disease. So in conclusion, liquid biopsy can provide clinically relevant information. The assays need to be validated and harmonized. Interventional clinical studies are required to demonstrate clinical utility of liquid biopsy. And I think this is very important. We need to do these interventional trials to really get the liquid biopsy accepted in the clinic. And interventional trials means that we change our patient management based on the outcome of the blood test. So at the end, I would like to thank the network here in Hamburg that I've established over the last 20 years in a network between research institutes and clinics treating cancer patients. And I'd like to thank the colleagues at my Institute of Tumor Biology. We are around 50 researchers from 14 different countries, a little bit like United Nations. And we get funded by the European Union quite a bit with 2 ERC grants, but also by the national funding bodies. If you are more interested in liquid biopsy, next week or the week after next week, I'm organizing and chairing a liquid biopsy major symposium at the AACR annual meeting in New Orleans. And this will be a very nice event. And if you have particularly young colleagues, please let them know that we also have a liquid biopsy Gordon Research Conference in June. And this is 5 days of liquid biopsy and a very good situation to interact with each other and talk 5 days about liquid biopsy in a small college nearby Boston. Thank you very much for your attention. And if you have some questions, please let me know.
Thank you, Dr. Pantel. I would like to invite Dr. Jamkar to come and take the question answer session, please. Thank you.
Dr. Pantel, I think it has been very brilliant and fascinating session. And everybody here get stimulated about your session. I just want to start my question because I'm a clinician, I'm a surgeon. So I'm interested in clinical applications. So what are the so far results of genomic panel sequencing with NGS with circulating tumor cells so far?
Well, I think that for the NGS analysis of the circulating tumor cells, of course, it will be interesting, or it's interesting to look, for example, at EGFR mutations, in the context of lung cancer, or KRAS mutations in the context of colon cancer, or look at HER2 amplification in the context of colon cancer that has relapsed, These are just a few examples, I would say, where you can get genomic information or PI3 kinase mutations, estrogen receptor mutations. These are just some examples. We published on these examples, and this is now doable. Yeah.
Video and audio signal.
Pantel.
Some point I'll dig out another cable and see if that solves it. If that doesn't, then I'll order you a set of powered external speakers.
There is somebody talking in between, right?
So, any questions from the audience?
Dr. Pentel, this is Dr. Sharma. Again, I'm a surgeon oncologist. We see the early cases all right. But obviously, all the early cases do not have the same prognosis at the end of it, and we are in trouble. So the question is, and you rightly said that the most important point which we are unable to assess is the circulating tumor cells in the brain. How far have we succeeded in sort of labeling them? And secondly, how far have we succeeded at the moment to assess the actionable drugs which can cross the blood-brain barrier? Though you may theoretically argue that if there is already a disease in the brain, you have already broken the blood-brain barrier. So how far we think we have achieved now, and what entails, right? You already justified. But at the moment, suppose I have a patient tomorrow, what do? I can't make out the patient has any brain disease, but she has some symptoms, and the CSF also is negative.
Yeah, very good question. What we are able to do today is we can find in approximately 60% of patients with brain metastasis, we can find circulating tumor cells, not 100%, right? We have some, let's say, false negatives, But in these patients where we find these cells, we can then further characterize these cells at the genomic level or at the expressional level. For example, if you want to know whether there is a activating mutation for EGFR, or if there is a KRAS mutation, that might block things, or if there is a mutation in KRAS where the new inhibitors could actually be actionable, or a PI3 mutation, So I would think that this now opens a new window, because otherwise you're absolutely right. It's very difficult to get access to brain metastasis, right? I mean, even if you know there is one, it's not easy to biopsy it, Mm-hmm.
Thank you. Any more questions, please?
I just have one. I have one. I'm on the screen.
Please.
Hi, class. It's wonderful talk. I'm just curious. Done a number of studies in patients. So when patients get onto a therapy, such as, let's say, chemotherapy, their immune system is highly affected. So have you guys compared CTC count, or either quantity or quality of CTCs, between the patients who are naive and then the one who are under treatment? Are there a difference in quality or quantity of the CTC between these two scenarios?
Yeah, I think both. We definitely see that the quantity of CTCs is going down during chemotherapy. So if we, for example, compare the number of CTCs in treatment-naive patients to patients with ongoing chemotherapies, in breast cancer, for example, we definitely see a decrease in the CTC counts. Of course, not in all patients, but in general, okay? And if we then look at the CTCs, and that's actually also the CTCs where we have now the cell lines derived from, which is interesting. we see that they have actually specific survival pathways, you know. They also go a little bit in the EMT direction, which may protect them from the action of the chemotherapy. Also, it's still a bit of a debated issue. But we really, I mean, can learn now really from this comparative analysis, we can really learn now what are the survival mechanisms, you know? I mean, how we have actually done that already in the bone marrow many years ago. We looked before and after chemotherapy, you know, and we were very much interested, what are these tumor cells after chemotherapies, the survivors? And in these old days, we could find, for example, that they had a very low Ki67 ratio, very low proliferation rate, right? So maybe the non-proliferating ones were the survivors, and that would make sense in the context of chemo. Yeah. Mm-hmm. Very nice seeing you.
Thank you. Yes.
Dr. Vikas, this is Dr. Ramesh from Chennai. My question really is for minimal residual disease when we do in a solid tumor post-treatment, when all the other parameters, including PET scans, everything clinically negative. What is the today the impression on the value of these or prognosis for these? Do you have any studies in long term?
Yeah, I think there are studies from our group, but also from other groups that have shown that if you detect the circulating tumor cells in the MRD setting, you know, or the circulating DNA in the MRD setting, that, you know, you have a higher chance to get an overt metastatic relapse. And the lead time can be a year or even more than a year before you detect the metastasis by imaging procedures. So our hope is now in clinical studies to use this lead time and treat the patients maybe earlier, okay? But this has to be shown really in clinical studies, whether an earlier treatment and also what kind of treatment will prevent then the metastatic relapse in these patients. But the prognostic information and on the detection of MRD is becoming stronger and stronger, I would say, from both using CTCs and using ctDNA. The key question is now, can we change the course of the disease? Huh?
Yes, that is one. But more than that, the number of CTCs, would that make a difference? What I'm looking at is in the long term, does the body immune system will take care of some of these cells which we didn't know before they existed or not? Now we could see them. In the past, we didn't.
Yeah, of course, the immune system plays an important role. And of course, we're also looking at immune checkpoint and immune markers, like I outlined that in my talk. It's very important to do so. But I should also say there are not so few cells. We calculated the number of cells which are in the body in an MRD patient, for example, in breast cancer. And we came up to something close to 1 million cancer cells in the total body. So even if you don't see anything with imaging, there might be quite a tumor load already in the patient. So it's not just a few cells, you know, that the body is fighting. The body is able to fight this cell number for quite a few years. In breast cancer, it can take 10 years or longer before you get a relapse, right? So if we understand more of the immune system, how it controls these tumor cells, that would be fantastic, and it's also part of our research program.
Thank you. This is Dr. Somashekar from Manipal, Bangalore. I have two questions. How do you compare CTC versus ctDNA? If we have an option of doing both very well, quantitatively, qualitatively, what do you prefer? One over the above or both together is better? Number one. Number two, we currently know that if you have a tumor block, doing a NGS on that is far superior than a liquid biopsy. Have we reached a level where liquid biopsy can replace need for any tissue ever?
Well, I think the first question is probably, you know, yeah, I mean, it depends what you want to know. Let's say you want to know a specific mutation, a BRAF mutation in melanoma patient, for example, right? Make it simple, BRAF V600 mutation, just a single mutation. then it's probably easy to do the DNA, you know. But in principle, it is both the CTCs and ctDNA, they deliver complementary information. You have some patients where you find the DNA and some patients you find the CTCs. It's not always the same, so you get additional information. But if you want to know, for example, the expression of PD-L1, whether the tumor cells have PD-L1 expressed or not, you cannot do it on the DNA. You need the CTCs. So it depends really on your clinical question. What kind of therapy and what kind of target do you really have in your mind, you know? And what kind of drug is really available for your tumor type? So I think it's really very complementary to look at both what the CTCs can really do for your diagnosis and the ctDNA. The CTCs, of course, allow you the analysis at all different levels, at the DNA, the RNA, and the protein, which of course makes a more enlarged diagnosis possible. Yeah. Mm-hmm.
Thank you, Dr. Pantel. There's an online question from Dr. T. Raja. I'll ask him to. There's an online question from Dr. Ramesh Khanna. Can it be used for a screening of general population?
No, I would be a bit careful for the screening of the general population. I think that the circulating tumor cells are not really in super high quantities in an early stage patient, but the same is true for all the other analytes. So I think for very early, I mean, for screening a whole population, you know, you need a very accurate method. Otherwise, you have many false positive and false negative findings. So I would probably not use it for a general screening. You may look at it if you have a risk population. So if you have a targeted risk population, let's say patients with some genomic background of a familial history of cancer or something like that, you know, maybe you start at such a risk population of individuals, you know, and see how many of your patients are then positive for the CTCs and whether you can use that for the monitoring of high risk populations, you know. But for general cancer screening, I think to me that is the second step, you know.
Thank you, Dr. Pantel. I'll invite Dr. T. Raja to ask the question online.
Professor, thank you, Professor. It was an excellent lecture. In fact, the question that I wanted to ask, you had just alluded to, Dr. Somshekhar also asked in similar lines. When we speak about liquid biopsy and its usage, I think we are using interchangeably the CTC and the ctDNA. And now the technology is moving towards RNA and exosomal RNA and so on and so forth. The question is, some genetic aberrations are notoriously well detectable by liquid biopsies. Some are inherently not good to be detected by liquid biopsies. For example, you know, fusions and translocations. You know, among the aberrations, some are well detectable and some are notoriously difficult to detect by liquid biopsies. Therefore, what is the current consensus, or even if it is not a FDA level approval, what are the broad guidelines approval for, you know, using these versatile technology? Thank you, Professor.
Yeah, well, I'm part of the ESMO, the European Society for Medical Oncology Committee on the guidelines for the ctDNA. So I was part of the committee. We have just finished our guideline paper, which will come out in Annals of Oncology. So there will be the guidelines published on that. We are also currently putting together the guidelines for the CTCs in our European Liquid Biopsy Society group. You know, we have a lot of competence here too. And we are, I mean, definitely the field needs guidelines. It needs also practical guidelines for our clinical colleagues. So our European Society is really very much into this for the next couple of years to really help guiding the field and help guiding also and showing also what you can do and what you cannot do, and also use the right method for the right clinical question, right? This is very important. So we are in the process of publishing these guidelines, and I mean, any of you are very also well invited to help us or give us feedback, right? Because we need always your feedback to make the guidelines even better.
Thank you, Dr.
Thank you, Professor. I have one more question, sir. Sorry, one more question. You know, I do have a patient. Just a couple of days ago, I see a patient. You know, as your case, lapatinib study showed, breast cancer detected 10 years ago, which was HER2 negative, and now has an isolated brain met and is HER2 positive. And then, you know, we have gone ahead to ask for a liquid biopsy to explain the, you know, I would explain to the family that there was probably a heterogeneous population early on. And so, and also in answer to Dr. Somsekhar's question, the liquid biopsies can detect heterogeneous population as versus a tissue biopsy, which can detect D mutation at that specific needle test. But in case, if there is a discrepancy between a tissue biopsy and a liquid biopsy, if it is done simultaneously, and the liquid biopsy shows more, you know, more elaborate or versatile changes, how do I act on that? So is there any way you can help us clinically decide, Professor?
Yeah, I guess that the regulatory, let's say the clinical guidelines, most of them are based on the tissue evaluation, right? But as you outlined already very well, we see sometimes in the blood that there are other locations, so we see heterogeneity, right? And we know with the clinical imaging, we don't see all the small metastatic lesions, right? So if I would see an additional genomic aberration and I would have an additional drug, you know, that is targeting it or can be used, I would definitely keep that in mind. I mean, there's no question. I mean, there's no debate anymore at the biological, let's say, conferences on heterogeneity. Heterogeneity is really something we should keep in mind. It drives us crazy because we don't like it. We want one target, one drug, you know, at a time and sequentially, and we want to make life simple, and medical life has to be, of course, also practical, right? We cannot do everything. But heterogeneity plays a big role as escape mechanism, you know. If you have different tumor clones, one can always escape if you just give a single therapy. And we know that already for many decades, right? That's why you have multi-chemotherapy, polychemotherapy, and not just one drug, right? So we learn now the same for the targeted therapies, and liquid biopsy can help to describe this heterogeneity.
Thank you, Professor.
Thank you, Dr. Pendle. There's one more question. We'll get this separate. One more question from the audience.
I'm Dr. Anuradha, coming from Tata Cancer Hospital. I had similar question which has been answered by you. It was asked by Dr. Somsekhar and T. Raj as well. Whether because CTC and ctDNA, they come from the liquid biopsy is the main origin. So whether they act as a rivals or the partners in case of cancer detection or for monitoring the disease. That was, I think you have already answered that question.
Yeah, I think I did that already. It's a very good question, and yeah, I can only repeat my answer, you know, but I hope I could make that clear that you can really profit a lot from the CTC analysis, and you have the ctDNA as a complementary tool. And I guess that in particularly monitoring really changes, you know, this should be really the key. I mean, sequential blood analysis are possible and easy, but you cannot do easily sequential needle biopsies, you know. So we have to understand how the tumor escapes and reacts to our therapy. And we can now measure that with a blood test. And I think this is a great advance.
Dr. Pantel, because there's a large audience online, there's one more question from Dr. Mohan Menon. How do you differentiate between driver and passenger mutations or mutations? Display driver and passenger mutations or mutations seen in tumor cells versus blood cells.
Well, I think the difference between the mutations in the blood cells and in the tumor cells can be done by sequencing also in parallel the blood cells, okay? For example, this is of course important to exclude the SHIP mutations, which of course need to be excluded. This and the driver and the passenger, I think when I see, let's say, a certain mutation increasing over time, you know, I mean, if I monitor and take sequential repeated blood samples, and I see that there is a mutation rising exponentially over time, you know, I think I have quite a good idea that that is really a mutation driving the disease, you know, and driving maybe the resistance to the therapy. That's also why taking repeated blood samples and doing repeated analysis gives you really nice additional information on what are the aberrations that really drive the relapse or drive the progression.
Dr. Pandal, thank you. I profusely thank behalf of organizer for spending so much of time and giving us so much of very intellectual, what is called brainstorming. Permit us to circulate your email ID so that all the audience can directly contact you about their questions.
Sure, absolutely. I'm happy to answer that also by mail. And thank you for giving me the opportunity here to present. And I also wish you a wonderful meeting. And I hope that one day I can join you in person in India.
Thank you. Let me invite our next speaker, Dr. Shafi Grover. He's going to talk on comprehensive genomic panel, liquid versus tissue biopsies for NGS set. Let me take this opportunity to compliment Dr. Jayan Khandare for this brilliant work of establishing the biggest lab in circulating tumor cell, that is One Cell Diagnostics. 3 years before, while I was visiting his lab, because I work as a chief medical officer with Index Technology under the guidance of Dr. Mohan. So I just asked him a question, can we combine this together? That doing an NGS sequencing of the circulating tumor cells and its impact. I'm so happy that this whole thing was taken by him. And I think today's exposition is a brilliant outcome of that discussion. So let me introduce Dr. Shafi Grover. He's one of the brilliant scientists that we have. And he has been working on NGS technology with genomic panel. And I think I'm very happy that the cost of NGS paneling that the foundation charges would be reduced. And at the end, as a clinician, we're interested in giving this latest technology to our people at the low cost. And maybe will be available through this technology. So I think I won't come between Shafi and you, Dr. Shafi.
Thanks, Professor Jamkar. And it is my pleasure to present you what we are doing here. So cancer is a complex disease, and it needs multiple technology in order to address this complex scenario. Thanks to technology, last decade, what we have seen, the precision oncology, we have seen a great achievement. But the question is, what is precision oncology? Precision oncology means personalization. And when we say personalization, and it has to be from the beginning when we get a sample from a patient. What we see is, as of now, is a generic analysis. That's the biggest challenge we face in the field of genomics. The general approach, what we have as of now in genomics is that every sample is run through generic workflow, and then generate the report, and from that we try to personalize the treatment. What is personalized analysis? This is what we do here. So it depends on starting in balancing the quality parameters and optimizing the entire workflow from sample acquisition till report delivery, and then you can personalize the treatment. So in order to optimize the outcome and minimize the sample failure, which we have been seeing in the last 10 years, that it's up to 30% of the time when sample is sent to the NGS lab, and sample, you get to know that it fails the quality parameters. The cornerstone of the precision oncology 2.0 is to treat each patient sample in an individual fashion so that you can personalize diagnostic workflow. So the challenge what we face today, we know very well the tumor is a heterogeneous, and most of the time when we get the tissue sample, the quality is so pathetic, and which is the reason that it fails in the first step of NGS workflow. Why? We know that tissues, first of all, it's an issue. We get a standard biopsy or we get a fine needle biopsy, and it's small amount of tissue, and there is a plethora of pathological tests to be performed, and you hardly left with any tissue. And in addition to that, what we see is tumor is heterogeneous, and most of the tumor samples have about 20% of the tumor cells. And if we look at the 20% and do the NGS, if we have a heterozygous mutation, means that one allele is changed, there is 10% of that allele fraction you will have. But being tumor heterogeneous, there are multiple subclones, and it goes below 5%. That brings us the question, how to address this. And that's why it becomes important to have a personalized workflows. Because we know that every patient's tumor is not same. We have been looking at the samples, DNA comes, the DNA quantity, DNA quality, integrity, it varies from individual to individual. And if it varies, then why we have generic approach? Why do not we personalize it in the beginning in order to minimize the quality failure and maximize the outcome? Another challenge what we face is the tumor is evolving. We know it very well. There is a very beautiful study. What you see here is, it was done longitudinally, where the samples, both tissue as well as liquid samples, were sequenced, within 2 weeks, less than 2 months, and more than 6 months. So what this shows here is, if your tissue is older than 6 months, you miss 40% of information. Just now, Professor Pantel and the great speakers were here discussing about the variation what we have from tissue biopsy versus liquid biopsy. So 40% of information is a huge. We cannot afford that to lose that. So how to address that? Again, the question comes, we have to personalize the workflows in order to get this. And this also suggests us that we have to longitudinally monitor each patient. And what we know that the tumor at the baseline, and when primary tissue is obtained, you may have some mutations, but we do not know after 1 month or after 2 months, as we see here in 6 months, they just completely changed. So how to capture that? So it becomes very important to determine mutational burden from the baseline, and then continuously follow based on your practice, based on your the observation you see in the patients, maybe 2 months, maybe in 3 months, and longitudinally follow over the year to see how tumor is evolving, and then how that you can address it. It can also help you to identify minimal residual disease. It can help you what could be the acquired resistance, and it maximizes actually success, clinical success, and ultimately patient benefit. So what OneCell DX, we have come up with OncoIndex test, which can be done at the baseline, run on the tissue biopsy, and followed by multiple liquid biopsy to address this issue. So how we have designed it? There are a lot of oncologists ask questions. I may need only 50 genes to test. Some say 10 genes are sufficient for me. But we know the cancer is a complex, and what we have seen based on the literature, based on the data analysis we have been working on, and also discussing with key opinion leaders, getting their opinion, we have customized and chose those 600 genes which are very relevant. And not only those genes, just select a gene and what you can identify it. We know in cancer, there are multiple genomic alterations which are very important, either driving the disease, either they are responsible for resistance to different therapies. So we have carefully crafted, identified 50 genes which are involved in fusion. As just now we were discussing here, it's very difficult to identify fusion, particularly from liquid biopsy. But our design has been made in such a way that you can capture fusions from liquid biopsy. How? We have designed the probes and primers in such a way that at least these 50 fusions, which are very important in oncological practice, can be captured, and including 35 introns, which are partners of certain fusions. We can capture insertion, deletions, important type of genomic alterations apart from single nucleotide variants, and we can capture amplifications, at least 25 important genes which are very important in terms of amplification. Most importantly, loss of heterozygosity. It's also very important. Genome-wide loss of heterozygous can be detected by this, as we have incorporated in our assay that those single nucleotide polymorphisms which can help us to determine genome-wide loss of heterozygosity. And it's one of the important feature, if you are looking for PARP inhibitors. Tumor mutation burden, as the panel sizes of big, 3 MBP target region, we can easily identify tumor mutation burden with more than 95% correlation to the whole exome sequencing. We can also identify microsatellite instability within a single assay. So this can be run on both tissue as well as liquid biopsy. So this is a snapshot of some of the important genes in leading those cancers like breast, bladder, ovarian, thyroid, melanoma. Some of the important genes we typically look into are already covered, and many more. So what is very important here is, and just workflow, as I was talking, the personalized workflow. And we have expertise in-house while we design these assays in such a way that we capture all these alterations which are important for your practice. And some of you have seen our lab, what we have built here, which is international standard lab. And most importantly, what I want to focus on is the two pieces of information, which is analytical interpretation and clinical interpretation. So this is where most of the oncology space travel. You get a PDF report, and whatever we have identified, and that's what you have to live with. Or you have to take that information, go to the Google, search it, what does this mutation mean? And how it has come into that report is a big question. So we provide both analytical interpretation as well as clinical interpretation. I'll be in a few minutes, I'll be showing you how we do it. We have built very nice AI-enabled analytical software which can help you to identify and better interpret the mutations you have in your report, and how it can help you to differentiate in terms of which therapy should be prioritized. So what this assay is, as I said, it targets 3 MBP sites, and it can be run on both tissue and liquid biopsy. And for a tissue, we need 1 to 3 FFP blocks. And for a tissue, we need 2 10 ml packaging tubes. And minimum amount of DNA which we can go for is 10 nanogram of DNA, and we can do the sequencing. And what we do is very importantly, for a liquid biopsy, we do as deep as 5,000x coverage, in order to identify those rare mutations which are otherwise you miss it if you do it in a low coverage sequencing. And sensitivity, we have more than 93% sensitivity. Specificity, we have 100%. Accuracy is 99%. And this whole work has been done, validated on cell lines, reference standards, and clinical samples. So I just want to highlight one case study, which we did it. This is a 55-year-old male lung cancer with metastasis to rib. So what we have got is tissue biopsy from primary lung and tissue biopsy from bone, which was the rib. And at the same time, we collected blood sample. So as routine, if we go with the generic workflow, the QC parameters you see for a tissue biopsy, it has actually very low, it was the average quality of tissue. DNA integrity was low, and amount of DNA was good, but the quality was low. So we personalized the whole workflow and then run the NGS on these 3 samples. What we have identified is EGFR mutation in all 3 tissues, which shows the concordance. So we have the ability to detect mutations. It could be from a tissue, it could be from a liquid biopsy, or it could be from a secondary biopsy. And there were 2 mutations. An additional mutation was RAD52, which, the percentage of RAD52 is just 1.4% in non-small cell lung cancer. So apart from EGFR is well studied, you can have EGFR inhibitors for this, but RAD52 can help oncologists to, possibly this could be the eligibility for clinical trial for PARP inhibitors. In addition to this, we have 2 gene mutations, which is TP53 and Notch14. That again, give you additional information for prognostic information about that patient. So this itself suggests that the sensitivity specificity of our assay is high, and it can actually address the questions you typically ask during when you want to go for a genomic testing. And most important is financial toxicity and how quickly get tested. It becomes very important. So you have to treat your patient at the speed of cancer, and you should be able to get a report as quick as possible. What we know as of now, it takes 2 to 4 weeks to get a genomics report. And we made sure that the workflow is in such a way that we can deliver a report within 10 days. So that's how you are enabled to treat your patients at the speed of cancer. And financial toxicity is, it has been defined that, the burden on the patient, how much they spend. As we know that genomic tests are very expensive. And so what we have made it, looking at the quality we provided, comparatively economical. And if we see what we are right now paying, and most importantly, what I see financial toxicity is not paying more. Financial toxicity means that, if because of we think that, I just want to go for a 10 gene test and treat the patients. If there was an important mutation, not in those 10 genes, but other genes, which you miss, is actually a financial toxicity for that patient, because patient's life is more important than paying, small amount of money. So, is there any questions? I want to show you how we can enable you interpretation, both analytical and clinical interpretation.
Questions, please.
That is fantastic. Just not an academic question about the last slide, which is most important. Your NGS based on a block is based around 75 TAT10, superb, because most of the people would send to foundation or somewhere where TAT is 3 weeks and the cost, it. Now the counterpart of the liquid by business strategy is really not placed well. You are above 1 lakh rupees, TAT is still 10. The garden TAT is 5 to 7 days, even today. There is a psychological barrier for those patients I have seen of 1 lakh. Try to see, bring it down to 1 lakh, 95,000, reduce the TAT within a week. You must do that because it's all based indigenous here. If somebody can do sending a specimen outside, get it within 7 days, within India, why 10 days? So it's just a suggestion. I know a lot of things goes to this. First model, right. Second model, somehow is not good. Please bring it below 1 and keep the TAT 1 week. Don't go beyond that to be successful in long run.
Please introduce yourself.
Dr. Somshekar from Bangalore Medical Hospital.
Thank you, sir. Your suggestion is well taken. As I said, it's within 10 days. little bit, I want to be, in safer side, it will be 7 to 10 days. We will try to make it even within 1 week.
Shafi, I think what Shom said definitely matters because ultimately that's where the restrictions come from our society. I asked you a question informally before you gave this lecture evening. Ultimately, you do 600 or 4,000 or whatever number of genes, unless and until if it is, actionable. For us, it's black and white and all kinds of things. At the moment that the trials that are going on, or still on trial basis, the access for all of us is very, very difficult. And my suggestion to you is that preferably if you have those mutations which are available and which you find, and if there are certain actionable mutations, you highlight those things along with the report so that we can either approach the trials process by applying to them, because all the patients can't afford if the drugs are not available. And obviously, if the drugs are in pipeline, they are also going to make a difference. I'm sure in future, when you have practically 100 drugs for 100 diseases, then the things become very easy and now. But at the moment of time, numbers will increase more and more if we are pinpointing actionable mutations. Thank you.
Thank you, sir. I have, very beautiful AI-powered software, which I will show you in a minute, which will help you to identify how many genes are actually actionable and when you should order which kind of a panel, whether it's a 100 gene, 500 gene, 400 gene, it's a big question. And that will help you actually to decide in that. And second thing is what we believe is, yes, there may be 250 genes which are directly actionable. But if we go beyond that, there are more genes which are indirectly actionable. For example, if you find a gene which could help you to tell you that, because of this mutation, the drug A will not work. As I'll give you one example. We have a tumor mutation burden high and PD-L1 high, and you want to start with immunotherapy. And if you get a mutation, for example, JAK1, STK11, that will tell you that there is a possibility that this patient will not respond to immunotherapy. So now this STK11, which cannot be directly actionable, or JAK1, which is not directly targetable, can become indirectly actionable. That's how this comprehensive gene panel like ours is going to be complete. All genes are going to be actionable. And second thing is, when we are talking about tumor mutation burden, one of the important biomarker in immunotherapy, and if we go to small gene panel, we will not get that information. Similarly, MSI, which has been approved by FDA for all solid tumors, right now what we have technology is 5 markers by PCR or IST. But what we know very well, there are more than 7,000 unstable sites in the genome. And if we just go for these 5 markers, turn negative, doesn't mean actually patient is a negative, because the other sites I have not tested. And in our OncoIndex, this assay, we have more than 2,800 sites captured in this assay, so that when we do this MSI, it's comprehensive. Imagine testing for 5 sites versus more than 208 sites. So the power of this is further unimaginable. It gives you more information.
Maybe you show a demonstration.
Like to take one more question.
So there are multiple panels available today in the market. How do I decide which one to choose and why?
Excellent question, sir. My demonstration will answer that. We have built this, precision medicine, oncologist console, where what we believe that, oncologist is a captain in the cockpit, who needs all the information on his or her fingertips. That's where, we are going to do this. Yeah. I think this internet little speed. Internet got disconnected. So this, oncology console, what we have brought features, I will not have enough time to demonstrate each and every piece, but to give you a highlight, we have a test result. You select your cancer type, and it will give you number of genes which are, highly mutated in that cancer type, and then you can select how many of them are actionable. And then you can select which gene panel is better suited for your patient. Yes, it has come here. So this is the patient dashboard. As I said, oncologist is a captain in a cockpit who needs all the information on his fingertips. There is, lag. I think it's due to internet speed. Loading. So what you get it, the moment you log in this, you get a list of your patients which you would like to see on that day, and you can select your patient. You get all the information about that patient. Typically, what traditionally I will say, you get a patient who has a file, all the reports, all the treatment prescriptions are there, and you have to keep turning on the pages and pages. So all that is on your tablet with just a finger. Can we do it? And sorry for this inconvenience, but we will show you how actually it is working on the tablet. Meantime, if there are any additional questions, I would be happy to answer that. Yes, sir.
I know that every genomic firm would love that we do sequencing with you and use your AI to interpret. But just I want to look at two scenarios. Do you also offer service? The example I'll give you. Suppose you have a Watson for oncology genomic from IBM. So Raja has it. We have lot of institutions in 7 years we have published a lot in Annals of Oncology. So an institution which already has a licensed version of say IBM Watson for genomic. Can I just get a sequencing, take the whole file in VCF so my Watson oncology leaves? Option one. If we do so, what component price reduction would happen? Or second, there are some government institutions where they have a Illumina, they want to do sequencing, but they don't have an AI backup because a good data without proper interpretation is a dot data. So do you also take that into the second half and do you split service or all in one single fit? We have to take both from you.
this console, what we want every oncologist to use it, and it's not only necessary that you should use onco-index test from one cell, but we would be definitely happy to you to use ours, and you will see the difference. But if you have done it from other service providers, and that can be brought into our oncology console, which is, I will not say it's like equal to Watson, but it will give you all the information what you need. Features like, you would like to understand patient like this, it can identify that. So yeah. So what do you have, patient dashboard? It's not coming up. Yeah, so we always face problems with technology. So what you see is kind of a list of patients you have to see on that day. And what you do is just finger tight. You'll get your patient profile. And this is it. All the information, what you have traditionally on the file of the patient is in this. So what you can do is, simply, zoom in, zoom out to get all the information for your patients. So as you need, you can do that information. For example, you would like to know what is that treatment given to this patient, and you have the summary around it. What was the treatment given? What was the dosage given when it was started? You get all the information there. What test has been, ordered? For example, you would like to see if there was some scan done, like MRI or CT scan. You get view of that. What was the impression from the radiologist, and you would like to see report around it.
It's a rare disease with a very diverse
full report out of this. And you would like to see actually, typically what you see the film of radiological report, but you can get actually in the viewer that whole image in front of you, which is a DICOM image, and you can play around it and see how the patient is, what happens to that patient, and whether tumor has progressed, whether it has reduced. And you may have multiple time points to see all the images there. So you can compare how this patient has improved, or it has progress. So coming back to this, the question we were having is which panel I should order. Now, this is test wizard here. You have a button, just click on that. So what it will take you to the application where you have all the cancer types, subtypes, rare cancer types information, and it will take you, for example, colorectal cancer. You would like to know what are the genes which are important for these patients. What is the frequency of that? What you see now here, single nucleotide variant information, indels here. And then you can have amplifications. What are the different amplifications important, for this cancer type? And how many genes which have a fusion? So, and what you have also, as we were just now discussing, is therapeutic information. As you have highlighted, the ones in the red edges, so indicate that there are targeted therapies available. So now the question is which panel I should choose it. Right now, what we have onco-index panel to show you how many genes are covered as per the data you have based on the various projects which we have accumulated, telling you the complete picture of these are the genes and how many of them we capture. And it's possible to, incorporate other competitor, panels because their gene list is available, and we can incorporate that, so that will give you a complete idea, which panel is actually capturing most of the genes in your patient's cancer type. And second thing is, once you know that, okay, this is the test which you should order it, and you just select it, okay, I want Encore DX liquid 600 genes, save it, and it's done. And so test is ordered, and we'll get the information that that test is available. Now imagine I have now, for example, ordered test. When it comes to you, say I order liquid biopsy, results came, and I get the information about the liquid biopsy. I would like to see the report. So I just click on the report, and a report will pop up. So now you see the report, and you can go, all the information about this report is there, and you can turn on the pages and see what information is available. As you know, it's a standard PDF report, but what is very important is what we give is active actionable report. And here is an icon, it takes you our application, which is AI powered application, and give you active report. Now this is where it makes a difference. So all the time you get report like this, there are multiple therapies, targeted therapies available based on the different alterations. So what should I do? Which one I should use it? So this can help you. Say, for example, I want to know what was the information about you. Typically, you have to read about this alteration with respect to KRAS mutation, and how many patients have benefited. And all that information has been nicely curated and presented to you. As I said, it's on your fingertips. You would like to see, okay, this is interesting. I want to read in detail the report, from the paper where it has come, that information. It will take you to that publication from where this information has come. And not only that is, you get additional information here, in detail, like, these are the therapies for patient, in patient's tumor type. And you would like to read this description about the, how the drug is interacting, how it has been done, what is the overall survival, all the details. And you would like, in addition to that, you want to know gene description, what is the role of the gene and how that the mechanism behind that, you get that. You want to further drill down to the variant information, and you get that variant information. And it's always on you, what you want to see. You can hide it, you can show it whatever information you want it. So in addition to this, you get the information, okay, the drug which can work as a, this one, sensitive, you have it highlighted in red and green so that you can easily distinguish. You would like to know what are the adverse events, you just click it, it will give the list of what are the possible adverse events this patient may face by this. So this way, what we try to help you, you have just 5 to 7 minutes you are spending with the patient, because so busy schedule, how we can enhance your practice by providing all the information on your fingertips so that you can make that decision within those 5-10 minutes and actually personalize the treatment to that patient. And now how we come to this conclusion, why I have reported KRAS mutation or FLT3 amplification or ALK fusion. So can I go a little back? How I came to this conclusion is, I have here a button which can help me to explore the variants, find the QC, what was the QC report, or get that PDF report. So I just want to take you to the explore the variants. So this is very important, this becomes handy to you. How I have come to the conclusion, this is the mutation which is very important, and this is how how KRAS was selected. So what you see is, on the tumor depth fraction, it was 16% of that patients has the mutation, and what was the coverage there? 3,000, more than 3,000x coverage. That's humongous, and you can get more information about this. Typing that plus button, what type of mutation is it? Is a gain of function mutation. What is the frequency of this mutation in different databases? What are the other computational tools which can predict the functionality of this? In addition to this, you have other databases. What you see here is, you want to select it, what population database, or somatic database. What is the significance of this in Kilnawar, whether it's a benign or pathogenic, or variant of unknown significance. This becomes very handy when you get US and you need to take call whether you should take it for your patient, or you should just. You can filter it based on the variant allele fraction. You want to see, something which has not come, you were expecting some gene should have been there. So for example, NRAS, you were expecting NRAS gene, and you want to see why it was not there. So the alteration found in this gene, so that you get the clarity, those important genes which you were expecting, there's no mutation in that. So this is how we enhance. Even if you want to go a little deeper, you can even visualize actual genome sequencing in the genome browser, and that's IG, standard genome browser, which will give you complete actually BAM file. You might heard about the BAM files, how that looks like. This is the BAM file of your patient, and this is the mutation, what you see in the red. And you can see how many reads actually, how many times the mutation has taken place versus what's there. So this can help you and to identify whether this mutation you should consider for your patient to take actionability. Similarly, for copy number variation, the copy number variation, what is the number of copies that has been, this patient has been got, like 9 copies are there, whether you will act out. Sometimes you may have very high copy number that will give you, yes, you need to target that gene. And similarly for fusions, what fusions, what were the partner gene, that information you get it here in one single place. So I would like to stop here and see if there are any questions before I take you to other activities.
Does your report addresses about the compound mutations, like if both are driver actionable, like amplification and driver mutations? Does it address, because there the confusion lies with the medical oncologist, which one is to be treated first?
Yeah, so that's the reason. That's the reason, what we do is we provide actually all the information to you at your fingertips. See, how I define a driver mutation it's a very complex scenario, and it's time consuming. Yes, we provided based on the literature. You have all the information. As I said, you want to know about that gene, what this gene is, and then how that mutation has altered that gene. Whether it's a gain of function, loss of function. That's how it is. If I have a gene which is involved in a tumor suppressor gene, and gain of function will not help me there. So because if it's a loss of function, then it becomes important for me because it's an important gene, and loss of function becomes important. Similarly, if I have an oncogene and if I have a loss of function, it doesn't, it's not important for me. For that, I need a gain of function. That's how you will know, okay, it will drive the cancer. Second thing is, now the allelic fraction becomes very important. So if I have a allelic fraction, say for example, less than 5%, and it's possibly subclonal mutation, and how it will help me if I have 2 mutations. One is, at a higher fraction, I can target that mutation, and other one is an allelic fraction less than 5% or maybe 2%, and it gives me that information that possibly down the line in 3 to 6 months, patient will start showing the resistance, that if that's a resistant mutation. That's how this console will help an oncologist to understand, because I know this mutation is a resistant mutation, but the fraction is low there. And on top of that, we give you all the access to the various databases, ClinVar, Cosmic will help you what is the presence of that mutation in the Cosmic. Will tell you how important that mutation is. Hope I answered the question.
Thank you. You didn't still answer my previous question. Maybe I'll catch you in a coffee. The most important question I asked you didn't answer. The software you showed, similar software exists from Siemens and GE. We did a pilot project in Manipal group. People who have a lot of Siemens and GE, they gifted it free. It's a beautiful software. The main thing is it needs to be integrated with our HIS system and track, because the patient privacy is at point. All the data link is somebody who enters. So if you have the software, who enters that? Because that itself is a big clinical work. Clinician needs good information interpretation in short time. Already, you know, in India, everybody is overburdened. So are you going to enable this to institution, the software? Because it's a fantastic software. It helps other patient management. And like, does it has a user license to integrate with HIS and track? Otherwise, two people don't speak, you know, they keep fighting, both software people, and we suffer.
Yeah, excellent question. You know, definitely. See, first, our software it's a highly compliant software, and it has the ability to integrate your institutional LIMS in a system. And what we believe that, you know, it's not only what information is captured in the hospital. The patient has also a lot of information. You know, we provide the portal to patient as well. They can also, you know, upload their information, reports which may have been done from outside the hospital, upload that, so that, you know, it can be fully integrated with institutional system, as well as, you know, patient portal is available. Maybe Mohan, you would like to add?
Yeah, excellent question. This is really where we shine. Obviously, there's not a time and place, but ability to aggregate the data from multiple sources, ability to bring it all together, store it, archive it, retrieve it, view it, analyze it using the best of the best in class tools, okay, and then provide you the insights. That's the part of the platform. Underneath infrastructure, it's a cloud native platform, mobile enabled. It is also regulatory compliance, 21 CFR Part 11, HIPAA compliance, GXP compliance, as well as GDPR in case you're involved in some of the European trials. So a very scalable, very open architecture. This is probably one of the first which has an API, open API, where you can easily integrate your hospital systems. And the vision here was that multiple sources at different levels of expertise. You know, here in India, we see that not too many places have the EMRs, right? So the patients carry a lot of their stuff, and we provide them on the patient portal and ability so they can upload. Because we fully realize that doctors are the consumers of the data, and anything you can do to automate using some of the AI stuff, right? You know, this is what really the underneath infrastructure that will differentiate this platform. Sure. Right. Well, I mean, you know, we've had a great deal of experience with Watson, overprice, you know, overpromise and underdeliver. You know, a very good example was what they did in the MD Anderson. We do have NLP, absolutely. You know, we essentially, but we use it very selectively where it makes sense. Okay, we support both structured, unstructured, and semi-structured data. Fundamentally where we shine and differentiate, you know, ourselves is the ability to take heterogeneous, multimodal, multi-omics type of data structures and bring them together to do secondary, tertiary analysis. That's our core competence. Does that answer the question? Absolutely, and that's what we'd love to have you as a part of our study group. You know, this is really very interesting for us.
Dr. T. Raja has a question. Dr. Raja, sir.
Thank you, sir, for allowing me to ask this question. Once again, sir, excellent presentation. I must really appreciate what you are trying to bring to us in India. Partly to answer a couple of questions there, and one suggestion and a question. With regard to the analytical part, the bioinformatics analytical part of it, you know, as Professor Somshekhar mentioned, we were one of the centers that had the Watson for clinical decision making, but we were also the first center for Watson for genomics. It's actually an AI-enabled, you know, genomics software application that is integrated with our molecular lab. But I would say at this point, it is still work in progress, and even IBM Watson Genomics is not a complete product yet, and they still have plenty of difficulties in, you know, but it's a work in progress, and our center is one of the first center to put Watson for genomics with the molecular lab in the country, I would say. That's my feedback, but it's work in progress. But with regard to what you have brought, you know, liquid biopsy, I must say that I'm a real fan of liquid biopsy. You know, what you mentioned there are several applications. We have had clinical examples, real-time patients wherein additional detection of 9p21 or MTAP has clearly shown why immunotherapy failed in a certain patient. So I'm completely sold on the technology of liquid biopsy and the versatile use of longitudinal follow-up. Now we have several patients on longitudinal follow-up. So therefore, my suggestion to you is that when we do longitudinal follow-up, there is a real cost constraint on these patients. So my request to you is that at least when patients are put on longitudinal follow-up, would you be able to give a package so that they can finally, in part, address the economics of it? And I know the use of longitudinal follow-up, it's going to be really, really useful in most of the patients. That's my suggestion. And one another low-tech ignorant question is that there are trouble with liquid biopsy, sometimes in interpretation like clonal hematopoiesis, so on and so forth. So therefore, Foundation One, after a period of time, now they have routinely started to report their tumor fractions in the liquid biopsies. So do you have such quality parameters? And I'm also aware that such parameters are not uniform across the lab, and they are now trying to report. When I had a conversation with Foundation One, they themselves have kind of regulated it, and now they are routinely reporting. So do you do any such thing like that? And the low-tech part of the question is that, is there a best way to draw the liquid sample? For example, I'm just wondering, is it a good idea to draw it at a particular point in time, say at early morning or in empty stomach and a low-fat diet? Would that really improve the yield of and also try to avoid certain noises like clonal hematopoiesis or low fraction detection? So that's my question to you. Thank you.
Thank you, sir. And your couple of questions, I believe that I should be able to answer all of them. The first thing is longitudinal analysis, as a package. I believe that it should be as economical as you are sending your patient for a international lab. It should be as economical as you pay there for entire 4 biopsies. So what I mean that with that, if you send it outside, you're going to pay about 2.5 to 3 lakh rupees, and we should be able to give you at least 4 biopsies, 1 tissue, and 3 liquid biopsies at that price. So coming to the next question, your liquid biopsy has challenges, particularly CHIP mutations. So what we have done is we have done some analysis and identified list of genes which are prone to show the mutations due to clonal hematopoiesis, and that are in our software, and using this ML technology over it, and it will give you this when you hover the cursor over that allelic fraction, it will give you information, is this potential CHIP mutation, so that you are aware possibly this is CHIP mutation, and you can take your decision accordingly. And similarly, it's not only for CHIP mutation, it's also for germline mutations. Now, if you have a germline mutation, and you will get that information when you hover the cursor over that, this is possibly germline mutation. So this, our software will help you to a certain extent to address all these questions.
So you will be routinely reporting on your WAF and all that. So that will help us to.
Yes, sir. That's what I have shown you. When you get the actionable report, and you can go explore the variants, and you have all the information for that, those mutations. What is the variant allelic fraction? What was the tumor fraction? What was the copy number? And what is the percentage around it? What is the total depth around it? So all that is in in that system. It's all in your fingertips.
My last question is, TMB has always been a difficult, difficult entity to master, particularly with the liquid biopsies. And I heard that you also mentioned about TMB. It depends not just on it depends on a lot of, I'm not an expert, but how many reads and depths and so on and so forth. So are you confident about reporting on TMB in your liquid biopsy? You mentioned that, that's why I'm asking you this question.
Absolutely, sir. We are confident about reporting tumor mutation blood burden from liquid biopsy, what we call BTMB. And we have taken care, it's typically tumor mutation burden is an analysis problem. And we have taken care of that, how to avoid or how to remove the noise so that you get actual tumor mutation burden, which we report.
Thank you, sir.
Sir, it was excellent presentation. I have one question. One clinical scenario, we had one patient, that is a CA lower alveolus. We gave radiotherapy, chemotherapy, followed by residual disease. We did surgery. After that, our CT scan and PET scan normal. We advised putting him in follow-up. We didn't advise any tumor, any liquid biopsy. After 6 months, patient developed lung, second primary in the lung. This histopathology is adenocarcinoma. Previous histopathology was squamous cell carcinoma. If I advise them liquid biopsy, will it help to detect the second primary early?
See, it's a very good question and difficult to answer this question. But what we understand, tumor is heterogeneous and very complex. And like your scenario, PET scan, all that have shown it, there's nothing, but it appeared. So we also know the limitation, that sometimes the tumors are so less, it is not detected by PET scan. And possibly in your case, there would have been small subclonal cells in patient, and which were not detected by scan. And by the time, they become grow, and then it shows metastasis. And we also know that, the tumor is evolving. There are well-known examples. We know HER2 negative patients turn to HER2 positive patients. Okay. And similarly, your case, this is what tumor is evolving. And that's why this liquid biopsy is better approach, because, you get wholesome of everything. There could be a small subclones and you get it, and that will give you a better picture what is happening. And how you can interpret it is there now. I have a mutation, so mutation may not be that frequent in the disease your patient has, and it could be important. For example, lung cancer mutation is present in the breast cancer or in an ovarian cancer. So it's difficult to interpret that time. How come you get that mutation? But so this will give you some idea. This is very common mutation in this type of cancer. But possibly there could be tomorrow, that it could turn to that disease type. So liquid biopsy, definitely I see as, it is giving a lot of information, particularly scenarios like this.
Any more questions?
I think Professor Pantel has made it very clear, in his presentation, as well as in follow-up questions. So it all depends upon what is the clinical question, as he said. And I feel both ctDNA and CTC are complementary to each other.
Hi, I'm Dr. Arvind from Chennai. Yeah, I think glad to be here, and a lot of insights. Just to take off from where Professor T. Raja left, with regards to longitudinal follow-up, I just wanted to ask, how frequently do you recommend the longitudinal follow-up? And you share some case scenarios in which these things have changed over time, considering most of this is immune related. So I just wanted to have some thoughts about it. And also there was some mention also about screening perspective, right? I think Professor Claus also was talking about doing the CTCs from the screening perspective, especially in the high risk cohort of patients. I know that there are a few sort of players in the market who actually advertise this from the screening perspective. So how frequently do you sort of recommend the usage of this technology in longitudinal follow-up of high-risk patients in screening, and also in the monitoring of patients, like in some of the scenarios mentioned? Thank you.
Yeah, I believe oncologist is the best person to decide what should be the frequency of the liquid follow-up, liquid biopsy. But on average, what we have seen based on the literature, based on the studies, on average, they say, every 3 months, a liquid biopsy genomic test should be done so that you get a picture there. And as I shown in one of my slides, what they have shown it, more than 6 months, you have 40% completely change what you have seen in 6 months before. So 2 to 3 months would be ideal option, but again, it depends upon your patient's presentation, how patient is doing, and your clinical understanding about that, and a call can be taken. And with respect to screening, so as Professor Pentel made it very clear, so what I feel is, if you find a genomic, alteration in those germline genes, and then I would again say, it could be combined, like Professor Pentel said, with CTC to screen the patients. So we know very well that if there is a pathogenic mutation in BRCA1 or BRCA2, the chance of breast cancer goes up from 5% to 85%. So it becomes then wise to screen those patients with CTC so that you can capture it early as possible.
We can take one last question. Right. Thank you, Dr. Gaur. I would like to now involve, I'd like to call Dr. G.K. Rath and Dr. Nirmal Raut to come and do the unveiling of our test, please. Thank you. Thank you very much. That was a very interesting first session. So we now move in the liquid biopsy domain from ctDNA to CTCs. The next session is all about CTCs. The next speaker is Dr. Catherine Alex Panibier, and she could not be here live because it's her daughter's birthday, but she has recorded this speech for us. And I would like to invite Dr. Pandarkar to come and introduce the speaker. Thank you.
Thank you very much. So our next speaker is Dr. Alex Catherine. She would be speaking on circulating tumor cells, and she is director of the Laboratory of Rare Circulating Human Cells in France. To her credit, she has more than 70 publications, 10 book chapters, and multiple awards, including from American Association of Cancer Research. So I think her topic will really enlighten us on CTCs. Thank you.
Well, thank you very much for this very nice introduction. First of all, I would like to thank Aravind and Vasudevan for this very nice invitation to be part of this conference on precision oncology reimagined. I would like to focus on liquid biopsy, the promise of CTCs. If you see this cartoon, you see that as soon as you have a primary tumor, even a small one, you may have more aggressive cells that are able to be released actively by the tumor place. They will reach the bloodstream, and of course, they will be attracted by different distant organs, like, for example, in breast cancer, you have the lung, the bone marrow, and the liver. And what has been shown recently is that in a very active way, these cells can be released as well from the metastatic sites. So mentioning the real-time liquid biopsy, it's really a non-invasive blood sample where you can capture and isolate CTCs coming from different sites, primary sites, but also distant sites. And it's very important because then you have really an assessment on the CTC heterogeneity, the tumor heterogeneity, because it's coming from different places and the more aggressive ones. What is important, you will see some example, is that you have frequently discordance between the genotype and the phenotype between CTCs and primary sites, tissue biopsies. So it's quite important to say that the liquid biopsy is really complementary of what you can get as information from the tissue biopsy. What is important as well is that you can repeat the liquid biopsy over time and during cancer progression, during I would say treatment of the cancer and so on. So it's quite important to mention that. And of course, you would have put the needle just a bit on another place, you might have had some different information. So again, you can really check this heterogeneity and mostly to assess and to detect minor clones that have been missed sometimes in the tissue biopsy. So quite interesting to see that and to talk about this personalized medicine or precision medicine. What I would like to say with this slide is that we really wondered whether there is one key step in the metastatic cascade. And using the Drake equation that we modified and adapted to the metastatic Drake equation, a mathematical model, we used a simulation on breast cancer to investigate the contribution of each step to the metastatic cascade. And interestingly, you see here that the most critical parameter governing the formation of clinical metastases is the survival duration of CTC. So very important to study this liquid phase of the tumor. The liquid biopsy is really the key to understand and to try to decrease this CTC survival to maybe be able to act on this metastatic process. What is important when you talk about liquid biopsy, you see that you have access to many different circulating biomarkers derived from the tumor, but also the background of the patient, which means that you can have access also to the immune cells. So it's really important to study these days also the circulating tumor microenvironment on the top of the tumor-derived biomarkers. So very important to see that as a great potential. What is important these days as well is that in many cases, you can have clusters or microemboli. You have like homotypic clusters, only CTCs, as you can see here, but you have also like heterotypic clusters, which means that you have a combination of for example, here neutrophiles and CTCs. And in this case, the neutrophiles are not destroying the CTCs, but they are helping, collaborating to help the cells to survive better, to proliferate better, and to self-renew. So very important data on that aspect. And it's really also a great work that Nicola Aceto is following for some years with good, I would say, good project and work. So the idea for the CTCs is really never travel alone, and they are helped by the blood microenvironment to survive better. Another very important point, at least in our lab, is really to try to identify the metastasis competent CTCs. And of course, it would be the Saint Graal to have access and to identify this subset of CTCs. Different ways to do that. If you want to expand CTCs, whether it can be in vitro or it can be also in immunodeficient mice where you can try to initiate xenografts. So quite interesting to see how you can perform that. I must say that it's really a challenge. And just to mention that we are the only group having today some CTC lines in colon cancer, and of course the only one with a series of 9 CTC lines from a patient who had metastatic colon cancer. So you can see that we could really get the expansion in vitro of CTCs, colon CTCs, before the initiation of the treatment. And you see again this nice heterogeneity among the CTCs, big cells, small cells, clusters. Then the patient was treated, first line in blue here. The patient really responded. So you see a decrease of the number of CTCs detected with the cell search. Again, heterogeneity among the morphology. But here it was not possible to have CTC lines. Here neither. But then the patient relapsed, and then we could get a very high number of CTCs in the blood. You see again big cells, clusters, and here it was possible again to get a CTC line. Then the patient was in a bad shape, not treated anymore, and just 1 week before the patient died, we could get 7 additional CTC lines. And you see here that surprisingly, we have no clusters anymore and mostly very small CTCs. So what we wanted to do is really to see, because it's a really unique biologic material that we have here, because we have CTCs obtained during treatment and also during the pressure of the treatment and cancer progression. So we want to know what are the last clones at the end that are the survivors. And it's really this very nice work that we published 1 year ago in Molecular Cancer. And to really summarize these data and to see the hallmarks of the CTC lines resistant to these therapies, I would like to mention first, they have a very strong stemness, which is not surprising because they are able to self-renew and to reinitiate some new tumors in distant sites. Second, they have a certain plasticity and they are able to go through the ENT, but also very quickly to go back to the MET. And I invite you to read this nice piece of work that we did in collaboration with Pelin from Turkey, but also with Mani from the MD Anderson, who will talk later today. And it's quite interesting to see this potential in these CTC lines. Of course, obviously, they have developed some tricks to resist to drug, and it's quite interesting to see as well that among the CTC lines. And I would like to mention this specific pathway, AKT-mTOR signaling pathway that seems to be very important in these cell lines. Of course, the metabolisms is very important, and xenobiotic metabolisms to get rid of the drug from the liver, but also a strong link to the mitochondria, and they are really hyper energetic. So we are still studying this aspect, that they are super CTCs, I would say. And for the end, I let this expression of ALDO-B, a specific proteins, tissue specific from the liver, as a liver metastasis source. And it's very important, I would say, because you see that you have 4 CTC lines at the end, just before the patient died, that could show that they have this ALDO-B expression. So they are really coming from the liver because it's a protein expressed really in the tissue, liver, hepatic tissue. So it's quite interesting to see that I mentioned that the cells can recirculate from distant organs and from the liver, for example, but here it's really a clinical validation that you have CTCs coming from metastatic sites like hepatic metastases. And it's quite important if you have, for example, a patient with a so-called localized cancer, and you can detect already CTCs that are expressing ALDO-B, you are sure that you have already occult micrometastases in the liver, which is very important for the clinician because they can treat the patient completely differently from those who have 0 CTCs, for example. So it's also an example where you can really take some real important information from the discovery at the biological level from CTC lines, for example, and to really help the clinicians and to translate directly in the clinic. So quite important. And also, we don't want to try to expand CTCs in all cancer patients because it's so difficult and challenging. We are less than 10 in the world to have achieved that. But I would like to say that we would hope that we can really identify a new pathway specific in this CTC subset that are able to reinitiate a new tumor in a distant site. So they are really 2 aspects here. First, they are specific subset of CTCs more aggressive than the others. at the origin of the cancer relapse in patients, but also it is, as we have rare events, it's also a way to get CTC lines, to have a way to have more cells to study, to work with. So the quantity is important, but not only, because it's not just a quantity of CTCs like that, but also the quantity of CTCs that are able to get to give rise to distant metastases. So very important. And I would like to put a very important note here, is that you have to keep in mind that the patients are not dying of the primary tumor, like in breast and prostate cancer, but they are really dying from their metastatic sites in more than 99.9% of the cases. So very important to know more about this metastatic cascade and also metastatic process, and to try to limit it, to try to make a patient maybe live with occult metastases, but to try to find a trick and to decrease the survival in the blood, like I mentioned before, or of course, to try to eradicate this very aggressive subset of CTCs that are metastases initiator CTCs. Now I would like to talk about the clinical applications, because it's really the key when you study like a new biomarker. Of course, it should get and give a benefit to the patient and to the clinician. So of course, we can talk about screening and early detection of cancer, and it's important to talk about the combination of different circulating biomarkers, mostly in early stage, and also to work on a high volume of blood. Second, in localized cancer patients, it's important to discriminate patients at low and high risk of recurrence. Also, in advanced stage of the cancer, it's important to have a prognostic information and to assess the risk for progression, but also to monitor on real time the new therapy. Very important as well. I would like to give you now example on two things. First, to identify therapeutic targets, but also it's important to have, for example, identification of resistance mechanisms. And second, I will talk about clinical utility of liquid biopsy to be able to introduce liquid biopsy in clinical practice. So first, let's talk about the CTC characterization. Today, I have only a short time to talk about PD-L1 expression on CTCs as a therapeutic target. So you see that in that slide, many tricks from the CTCs to evade the immune system. And today I would like to talk only about PD-L1 expression on circulating tumor cells. And you may know that these tumor cells may express PD-L1 to escape the immune cells. And by interacting with PD-1 on T cells, they are not destroyed anymore. So they are really in a camouflage. And the idea of the immunotherapy is really to break this bridge by giving anti-PD-1 or anti-PD-L1 antibodies and to reactivate these T cells to eradicate the tumor cells again. And in 2015, we were the first one to establish this test detecting CTCs expressing PD-L1. And it was really a success to have that because it was almost at the same time of the Nobel Prize for the immunotherapy. So very important to have also these circulating biomarkers to try to have this precision medicine in immunotherapy. So just to show you how it looks like. So we use the cell search system with a panel of CK8, 18, and 19. Staining, of course, the nucleus, CD45 negative staining because it's specific of leukocytes. And in the last channel, you see the staining of PD-L1 on a CTC because you have here a very nice merge picture to show you what is a CTC. Of course, in the same patient, heterogeneity because you have also CTCs that are not expressing PD-L1. But in this first work, we could show that more than 60% of the patients had CTC expressing PD-L1 in metastatic breast cancer patients. We went on and we added more subtypes of breast cancer. You know that you have different subtypes and you see the proportion of the different subtypes here. And what we could show is that first, up to 80% we could get CTCs detected up to 36% CTCs PD-L1 plus, but this time we could really also detect expression of PD-L1 in the tissue biopsy. And when we compared, we could show that first, no correlation between tissue and liquid biopsy for the expression of PD-L1. And second, only the presence of CTC PD-L1 plus was an independent biomarker for shorter progression-free survival. So the clinical outcome was given only by the CTCs PD-L1 plus. Another study in non-small cell lung cancer, advanced stage as well, more difficult to detect CTCs in that case, only 43.4% had CTCs, and even a less subset of patients with CTCs PD-L1 plus. But the key information again is that no concordance between tissue and liquid biopsy, and only the CTCs, and mostly CTCs PD-L1 plus in green here, could show a worse clinical outcome in these patients, talking about progression-free survival, but also overall survival. And when we looked at this PD-L1 expression in the tissue biopsy, not associated with any clinical outcome. So very important to show that this liquid biopsy is really associated with a clear clinical relevance and clinical outcome. And in this case, we could really analyze as well ctDNA and also exosomes expressing PD-L1, but I cannot present this data right now because they are still under analysis. So I hope I can say more in a soon future about that. And we are running these days a new Alcina clinical trial in the context of immunotherapy this time. And we will have also a follow-up before the treatment and during the immunotherapy. And again, we will have a huge biobanking, talking about plasma, exosomes, platelets, ctDNA, but also in the cellular fraction, we will get CTCs, but also immune cells information. So very, very important to have this full profile of the liquid biopsy. Another example is HER2/neu, and HER2/neu in breast cancer, you know, is really a very aggressive cancer, but hopefully we can propose a specific targeted therapy. And why sometimes there is a clear discordance between, again, primary tumor and the CTCs is that first, you have minor clones that can be missed by the pathologist in the primary tumor, and thanks to the liquid biopsy, you have this information. And second, it can be also that you have a gain of this HER2/neu amplification during the dissemination. Anyway, at the end, liquid biopsy can inform dramatically the right information for the metastatic sites that might be then HER2/neu positive. And of course, in that case, it could be of utmost importance to give the targeted therapy against HER2/neu. And it's really something I would like to highlight from our colleagues and friends from Germany, Sabine Casimir-Bauer, because she really proved, again, if you check in the primary tumor, like we did in the Merkel cell carcinoma, cell by cell in the primary tumor using the deep array, you can see that you have some minor clones that are HER2/neu positive and that were missed when you look the tissue biopsy as a bulk. So very important to notice that. And we are running these days a project to detect CTCs expressing HER2/neu in the cerebrospinal fluid using the cell search. And it's quite important to detect this leptomeningeal metastases. You see here on the left side some CTCs HER2/neu positive, and you see on the right side here HER2/neu negative. So we are still waiting for the last data, and one paper is under review. Then I would like to mention also the clinical utility, which is really the key to go into clinical practice. So this first clinical data here, the stick breast, meta-breast clinical trial was really important, an interventional one, where we had to choose between endocrine versus chemotherapy based only on the CTC number. So up to 4 CTCs, it was considered as a good prognostic, and then the patient could get endocrine therapy. And when we had 5 CTCs per 7.5 ml of blood or more, we could get chemotherapy because it was considered as a bad prognostic. So this clinical trial was really a success. It was done on more than 800 patients, metastatic breast cancer patients. And now in France, we have asked the high authority of health to maybe find a way to refund this test, because it was a medico-economic evaluation as well as a quality of life for the patients. So of course, and also maybe the state can gain money to treat better the patients directly than to have early relapse and then to have other costs, I would say. So it's really something that is really in progress. So we are still waiting for the feedback from this HAS, and we hope for the best. So you see, we need these interventional studies to go in the clinical practice. And we have another tactic project on prostate cancer, metastatic prostate cancer. And the data are now in the analysis as well. But just to show you what is really going on in France for the refounding. So when we talked about liquid biopsy, and Klaus mentioned that before, about liquid biopsy, it was really first a cell, at least a CTC, because it's like that. And now the new definition updated last year in Cancer Discovery is really all the different circulating biomarkers that you can find in the blood derived from the tumor, but also the immune cells or the fibroblasts and so on, and also not limited to the blood, but also in any kind of body fluid. So it's quite interesting to see that we can get really full of information from that. And to conclude, what is the future of liquid biopsy? First, to push liquid biopsy into widespread use, and in the clinic, more interventional studies are needed, like the stick metabreast I just presented you. Second, I hope that we can develop an algorithm in the future that can combine different circulating biomarkers to obtain a precise tumor profile. And of course, it can be cancer type dependent. Which biomarkers can be more informative in which cancer types, and also at which step, at which stage. Very important to define that in the future. And finally, we need to define guidelines and SOP for liquid biopsy, because we have too many viabilities for the moment in pre-analytical and analytical steps. And we need to rely on big consortia to achieve these goals, like the European Liquid Biopsy Society already mentioned before. So at the end, when you look at this Nature paper and you see during the last 20 years, what are the key discoveries, and you see that liquid biopsy is really nicely highlighted. And I believe that in a soon future, we will track better cancer in liquid biopsy. At the end, I would like to thank my collaborators, of course, in France at the European level. And I would like to mention Klaus Pantel for sure, but also in US. And I would like to mention Mani Sandurai, who is present in this conference as well. At the end, also the grants, because without money, we cannot work at the national French level, but also at the European level, and also my group, a very nice group working with me, not so big, but very dynamic. And I'm very happy to welcome people from Iran, from Mexico, from India, but also very soon from Italy and from Spain. And I would like to thank you for your attention. I'm sorry today I cannot be physically with you on live because it's the birthday of my daughter, but I'm really with you, and I hope you spent a very nice conference, and I hope to see you in the soon future. Thank you very much for your attention.
I'd like to thank Dr. Panavia for a wonderful talk. If anybody is interested, Professor Pantel, who spoke the first keynote, and Dr. Panavia, who are close collaborators and who have actually coined the term liquid biopsy, they have a wonderful paper on minimal residual disease in 2019 Nature Reviews. I would suggest that paper to anyone who would like to read more on combination of CTC and ctDNA in detecting of MRD. I would now like to call Dr. Sanjay Sharma and Dr. Ashok Sharma to please come on stage and unveil the OncoDiscover product. Thank you, Dr. Sanjeev Sharma. Thank you, Dr. Ashok Sharma. Now we have a very special person with a talk. Just take a minute. We have Dr. Pankaj Chaturvedi, who is our Prerana Stotra, who has been the greatest support to our research. And he has sent a quick bite for playing online. So I would request the team to play the AV.
Dear Jayant and entire team, it is indeed a matter of great pride for me that you have reached this stage. This is all because of your hard work, passion, and commitment. Let me tell you that I have never seen such a committed researcher and a social entrepreneur like you, and what you are doing is an amazing work. And certainly, it's an honor for Tata Memorial Center and personally for me to be associated with you. And I must acknowledge that the research that we have done with you and the work that we have done with you is going to be very path breaking. It is going to really disrupt the whole technology of circulating tumor cells, and now you are entering into one cell. If you remember, we recently published our paper on oral cancer of 152 treatment naive oral cancer patients, where we found that the presence of circulating tumor cells has a very high sensitivity and specificity for detection, and you could differentiate true positives with negatives from once we had 3.5 CTC or more. Now, what was really surprising and something that is not seen before is that a CTC that is more than 20 was indicative of nodal metastasis. That means we have come to a stage where by simply doing a blood test, we can know whether a patient has a nodal metastasis or not. What is more important that with elevated CTC with the cutoff of 13, what we found that the median survival was higher in those who had lesser CTC compared to those who had high. And also, the presence of CTC was strongly correlated with the established prognostic factors, histological prognostic factor, I mean, like perineural invasion and extranodal extension and depth of invasion. So that means that we have come a long way in understanding the head and neck cancer, where we always thought that it is a loco-regional disease. But by presence of CTC, even in early cancer, we know and we understand that oral cancer is a systemic disease, and therefore presence of CTC is going to revolutionize the treatment, and not only treatment, but management, early diagnosis of recurrence, and perhaps ability to escalate the treatment who have higher CTC. That means they have
Thank you. Thank you to Dr. Pankaj Chaturvedi for the beautiful bite. Thank you. I would now like to welcome Dr. Pendarkar to introduce our next speaker, my co-founder, Dr. Jayant Khandare.
So I think after great presentations, we have another very important presentation on OncoDiscover, first in class indigenously developed DCGI approved patient monitoring. So this is the presentation is by none other person than Jayant Khandare, who is CSO of One Cell Diagnostics. Dr. Khandare is head of the research of the MIT School of Pharmacy, Pune, and managing director of Actorius Innovations and Research, a Pune-based biotech and medical device company. He is a fellow of the Royal Society of Chemistry, UK, and has been the recipient of prestigious Alexander von Humboldt Award for Experienced Researcher Foundation in Germany. Dr. Khandare is a PhD in chemical engineering from National Chemical Laboratory, Pune, and multiple postdoc stints at reputed universities in US and Germany. He is also the innovator of the OncoDiscover liquid biopsy technology, which is India's first regulatory approved technology for detection of circulating tumor cells in cancer patients. He has published more than 15 papers. He has US patents, more than 60 high-peer papers in the areas of novel drug delivery. I think we here are honored to have him here and present us the onco cell discovery. Thank you. Go ahead, please.
Good evening. Thank you very much. It's a privilege to speak today a technology that's the first medical device that has been approved by Drug Controller General of India. We speak about innovations, we speak about revolution. OncoDiscover 2 is a first-in-class, indigenously developed, DCGI-approved patient monitoring test. The best work of the pathology of a cancer is done by those who are actually studying the nature of a seed. I think the three pioneers, those who termed the liquid biopsy, have spoken extensively about liquid biopsy and the role of circulating tumor cells. I think tonight we should understand the presence of a single circulating tumor cell in patient's blood is a telltale signature of a metastasis. And 90% of the patients die because of a metastasis. Therefore, there is a question whether we should ignore the metastasis. And the worst clinical outcome that Dr. Pankaj Chaturvedi mentioned, that the presence of a circulating tumor cell can predict the clinical outcome in chemo-naive patients. So it's a first DCGI-approved indigenously developed test that has actually taken off in this Pune center. Dr. Harshavardhan, Honorable Health Minister of India, launched the OncoDiscover liquid biopsy technology in 2019. We publish patents and publications. I have a great pleasure in announcing that today we got an email from ASCO that 5 of our abstracts have been accepted in ASCO. That would be a rare occasion for any Indian company and to the one cell which is actually doing this wonderful job. So as I mentioned, the considerable fractions of cancer patients have metastatic disease, and we know that the Indian patients succumb very rapidly, and the overall survival of 5 years in India is less than 30% compared to that of an European and Western world. Most of the oncologists have seen these images when patients come to your clinic. I think the whole body scan, in this case of a patient with a metastatic NHL is imaged. On the right-hand side, you will see a single cancer cell and its steps of an extravasation. Unfortunately, there are no in vitro mechanisms to prove that the cancer cells can cause invasion in human body. We have spoken about circulating tumor cells. I think in the paucity of a time, I would not repeat what do they mention, but CTC clusters are very aggressive and they can be 50-fold developing the metastasis over to that of a circulating tumor cells. The beauty of capturing circulating tumor cells, which are exceptional, low in abundance in a blood sample, is well established since 130 years. OncoDiscover is a tool or a technology with a kit which has an immunomagnetic bead, and at the periphery, the EpCAM antibody is attached in such a way with increased sensitivity and the specificity. On the right panel, you would see the beautiful circulating tumor cell in one of a breast cancer patient that we have imaged. We have developed an OncoDiscover liquid biopsy technology, which is proprietary, which has a multi-component systems, in vitro diagnostic kit, and that captures circulating tumor cell within 4.5 hours compared to that of 12 days as a TAT in US and Europe. Dr. Pankaj Chaturvedi already mentioned about the possibility of circulating tumor cells in Indian population, because most of the clinical studies with this kind of a technologies are evaluated with other countries. Dr. Prabhash is here today, and we have been learning extremely what clinicians want. Dr. Pankaj Chaturvedi mentioned in one of her tweets that this is an important step towards prediction of an outcome through a simple blood test. I would leave to the oncologist to come forward and evaluate this kind of a technologies and in your own clinical settings at different patients population at any stage of their disease. What has been established in the past, the clinical significance in the prognosis in breast cancer, colorectal cancer, and the prostate cancer, that the probability of overall survival with the patients having less than 5 circulating tumor cells have actually a shorter progression and the overall survival, which is about 18 months. The critical problems in cancer are known, that there is an MDR, there's a cancer dormancy, and minimal residual disease. Both Dr. Catherine as well as Dr. Pantel has actually mentioned these 3 issues and the problems. And the circulating tumor cells can actually come forward and establish 3 critical problems in our settings. Arvindan just mentioned about a minimal residual disease. I think this is the most significant manuscript or a review that has been published by Dr. Pantel, which speaks about the minimal residual disease and the time of a progression in the years. But often this kind of a progression is missed in the clinic because the technologies like OncoDiscover have not been accessible, affordable, and available into your clinic. OncoDiscover presents that possibility wholeheartedly in India now. So liquid biopsy clinical implementation and advantages are several. And as we have been talking about the complementary utility of circulating tumor cells and circulating tumor DNA. But let me assure you that the surveillance of a patient who are disease-free or they have been cured, the best possibility and the probability in detecting them earlier for their relapse or a metastasis progression is in the live cells, that is circulating tumor cell, because they are the carrier, they are the fugitives of distant metastasis progression. One of a large study that we did, maybe in world's largest clinical trial in Indian setting with 500 head and neck squamous cell carcinoma, me and my team published this recently with Dr. Pankaj Chaturvedi and Dr. Kumar Prabhash, and it indicated that the patients who had more than 13 circulating tumor cells survived for a lesser time over to the patients who had lesser circulating tumor cells. I think there is going to be a possibility of considering liquid biopsies, including circulating tumor cells, in various guidelines, as Dr. Catherine mentioned in one of her talk and the meeting with us. We monitor patients longitudinally, and in this case, real clinical scenario that the patients have been monitored by circulating tumor cell in 1.5 ml of their blood samples over 7 times. And there are favorable and unfavorable situations where the primary outcome and the metastasis progressions could be different. The best scenario that the patient does not have a PET scan, the patient responds to the treatment, and the patient do not carry any circulating tumor cells. That's a favorable component of this liquid biopsy. But in 80% of the time, in our 2000 patient sample analysis, we found this unfavorable. I leave it to the oncologist to analyze it. Dr. Nirmal Raut, who is sitting here, has used circulating tumor cell, has prescribed it often, and he tweeted this with a presentation of a case, female 65 years, cancer of a rectum, various treatment, and I think disease-free status, no positive presence for the primary tumor. For whatever reason, you might ask him that why did he actually prescribe this OncoDiscover test, send the blood to our lab, surprisingly and panically to the patient and oncologist that we found 2 circulating tumor cells in 1.5. What to do from here? I think there was a lot of discussion, and Dr. Nirmal decided to actually go for the metastatic possibility through the MRI in abdominal region. And on the right-hand side, to his surprise and patient's bad luck, there was 4 metastatic lesions that were found into the liver. So what does oncologist does? Comes out from the clinical trials and the stratification that has been actually significantly done. I'm sure that all the oncologists present here will appreciate the fact that the clinical trials cannot encompass all the objectives that actually are typically mentioned. This might be one legendary case that the patient actually is taken in SIRT and the CTC value after SIRT was 0. I think SIRT was done in Tata Memorial Hospital. If the patient didn't found circulating tumor cell, what would happen? Maybe you could talk to Dr. Nirmal as an patient oncologist. Case number 2, that the disease monitoring in Indian setting in head and neck and oral cancers. We know that the presentation of this case, particularly female 80 years, refused surgery, and the patient was taken to oral metronomic chemotherapy, very inexpensive, 3,000 rupees per month. Continued complete responses, as you see in the figure. On 22nd April 2021, our technology detected 3 circulating tumor cells, and we are probably not aware of the presentation of this case. Within few days, with metronomical treatment, and the circulating tumor cells came down from 3 to 1. And next 1 month, circulating tumor cells were actually 0. I think this is what is a beauty of a liquid biopsy that cannot be covered up. And both Dr. Catherine as well as Dr. Pantel accepted the fact, the limit of a clinical trials and the exploration what Dr. Nirmal Raut has done. I think there are many case studies where you could have this kind of presentation and the impact can be analyzed and the oncologist can take a decision to change the treatment if there was a presence of circulating tumor cells. Dr. Vinit Govind Gupta is here. He has also seen some of the facts which he would otherwise not have been explored. In the time limit, I would not cover up what is the utility of circulating tumor cells in the clinic. But the fact remains very important that circulating tumor cell is going to be the buzz in the next genomics area, where you could have single circulating tumor cell and do multiple components of a science, including genomics, transcriptomics, and proteomics. And please watch this space. We will be actually coming with this kind of a important omics data very soon. So accelerating precision medicine, CTC multi-omics biomarker data analytics will be powered by AI and ML. And Mohan and my team is here to actually explore these components from now to ever. Thank you for your support. But in the end, it's not just about taking a blood sample. It's about taking the despair out of a cancer. And I leave this power to the oncologist, along with the technology that will accelerate the precision oncology in this country. Thank you very much.
I would like to take some questions.
Sir, myself, Dr. Sasipriya from Ashamtra Cancer Hospital from Tamil Nadu. We have one patient, CA rectum. We gave preoperative radiotherapy and concurrent capecitabine. And after 6 weeks, we did CT and PET CT, everything normal. The patient also not willing for surgery. If this liquid biopsy, will it be useful? If the circulating tumor cells are 0, can I put the patient in follow-up?
Since I'm not an oncologist, I would take a liberty to pass your mic to Dr. Nirmal, who would be better person to actually answer this question.
There are no studies to do that, but if the circulating tumor cells are 0, then probably if the scans are not showing anything, you can keep them on follow-up. But you need to keep them on serial follow-up of CTCs. If the CTCs start rising, probably she is developing the disease. For 3 months, every 3 months for at least a year or 2. Yeah.
Thank you very much.
Jayant, I'm here to give you all my compliments and congratulations. I'm extremely happy that with all your work, you have made India proud. I'm not talking about food. And these claps suggest all the appreciation for all your efforts. I think we need few more giants like you for making India still a better place.
Thank you very much.
And I have a very question, sir. Maybe you might answer. So there is a patient of carcinoma breast, T2N0M0. Nothing on, say, metastasis workup, and he has CTC positive. What does it mean?
Sir, CTC positive means when has the patient undergone all the treatment and on follow-up?
Yeah, patient operated, everything done. T2N0M0.
So just one value of CTC doesn't give any correlation. What we have to do is serially, if their CTC is coming after 3 months, is coming down, then probably you can still observe. But if the CTC is rising, then probably you need to keep
So
it.
you don't call it metastasis.
No, but because the immune system can clear that metastasis also.
But there might be some immunological mechanism within your body to take care of CTC.
Yes.
Thank you.
What you could do is actually analyze this circulating tumor cell and look for the primary or the secondary difference by PD-L1, HER2 positiveness. We are validating this kind of a markers which will actually distinguish between the primary as well as the secondary cancers.
Anyone else? Right. Thank you, Jen.
Thank you.
Can I ask one question?
Sure, sure.
Dr. Jayant?
Yes, Dr. Raja.
Yeah, hi. Dr. Jayant, congratulations. Amazing work. Congratulations. I'm glad that I was speaking to you earlier. And I'm also glad, and I will also tell everyone that you have also supported one of our, or actually a couple of our patients in whom. And so my question to you and to Dr. Nirmal, such a dear friend to me, I'm glad to see him. The question is, one of our pancreatic malignancies, as you know, the person that I'm mentioning, after we completed all treatment, including chemotherapy and then radiation, then on serial monitoring, we did see the CTC 2 cells, and then we were religiously repeating every month the CTC, and over a period of time, exactly after the 8th month after stopping all treatment, we saw the cells disappear. So in fact, I was about to ask you, just by the cells being present and then over a period follow-up, they do disappear. And I think Nirmal indirectly responded that there could be other mechanisms which will probably eliminate. So this is something that I want to point out, that just by CTC presence, you need to monitor to see whether they are going up or down. And our patient fortunately did finally get rid of that CTC. And I must really thank you and congratulate you, and thank you, Nirmal. And what is your comment on this?
Sir, yeah, one particular value is not much of use. You have to do serially whether it is coming down or is it up. And so in this patient, it had gone down after 8 months. So that means that the CTCs were normal, and so the disease was taken care of.
So I must say that actually that should be a congratulations should return to you. So what goes around comes around, and thank you very much, doctor, for using actually and monitoring this patient for almost 7 to 8 number of times. So
right.
It's a very favorable outcome that can be actually easily done with a small pool of blood.
But congratulations, really.
Pleasure, sir. Thank you very much.
Jayant, I just wanted to add, have you seen the movie No Time to Die?
Yes, sir.
I think that is the further, can you extrapolate that whole health genomics to cancer genomics? Sir, have you seen that movie?
Sir, no, I have not.
Please see that movie and enjoy on genomics.
Okay.
And I think that we should
No
extrapolate
time to
for
die, right?
cancer.
Yes.
Thank you, Jen.
Thank you very much.
I would now like to introduce Dr. Raja Pramanik from AIMS, who has done an international study with us, to come and speak. Thank you.
Thank you, Aravindan, for the invitation. Ladies and gentlemen, very good evening to all of you. It gives me immense pleasure to be speaking with such an august gathering. And Over the next 10 to 15 minutes, what I'll be speaking about is the role of CTC in recurrent metastatic head and neck squamous cell carcinoma. And these are the data from the translational experience from a phase 2 study that we have recently conducted in our department. So to just give you a background, I will be just presenting this study in very brief, and then I'll go to the translation arm of the study, which was done in collaboration with Dr. Jayant. So you all know that head and neck cancers is the very commonest one in males, second commonest cancer overall in India. Advanced stage plagues this disease, 2/3 of cases are advanced, and majority of these relapse within the first 2 years. And until recently, we did not have a biomarker for this. So in the recurrent metastatic setting, what are the options for platinum sensitive disease? We have immunotherapy plus chemotherapy, or chemotherapy with targeted therapy, cetuximab. But for those patients who relapse after exposure to platinum, what options do we have? So we are left with single agent nivolumab or pembrolizumab, which is so costly and out of reach of more than 95% of our patients. So what we do is maybe a conventional chemotherapy or oral metronomic chemotherapy, either 2-drug or 3-drug chemotherapy of late. So this has been most of our practice. So we have been asking ourselves the question, can we do anything more for these patients using the resources we have at our disposal? And so we try to develop an indigenous regimen for this, for these patients, especially those who have a good performance status, not the bad performance status ones. And therefore, we developed this idea of converting the age-old regimen of CMF, that is cyclophosphamide, methotrexate, and 5-FU, to EMF. So you all know that cyclophosphamide, this is the regimen of CMF, injectable methotrexate and 5-FU, day 1, day 8, and oral cyclophosphamide for 14 days. And we repeat this every 28 days. So if we just replace this cyclophosphamide with erlotinib daily, we have a new regimen called EMF. So we tried this to experiment with this regimen. What this does is that for these good performance status patients, we give them a chance to expose to all the active agents which are present at our disposal. All these are inexpensive drugs. And there is a strong preclinical rationale to it, because methotrexate is exited out of the cell because of this ABCG2 protein, and erlotinib blocks this. 5-FU itself has a single agent activity. And this regimen is very hospital friendly. It can be given by push injections in the injection room. It appeared safe to our initial trials, and then it is very cheap. And IV ensures compliance, that the drug is getting inside the body of the patient. Many patients of ours are not compliant. They have a Ryle's tube in situ, etc. So with this rationale, we started this study, single-arm feasibility phase 2 study of EMF in platinum refractory recurrent metastatic head and neck patients. Very quickly, we aimed for a response rate that it will increase by 20% over conventional chemotherapy. Primary objective was 3-month overall response rate, CR plus PR. We had a lot of secondary objectives, overall survival, progression-free survival, quality of life analysis, and then we had this translational arm. We did cfDNA, not the ctDNA, the total cell-free DNA as a biomarker, longitudinally assessed it at 2 points. And we also assessed CTCs as a biomarker, and there we got the help from Dr. Jayant Khandare. So this is the study. We had the ethical approval, and the QOL analysis was done by EORTC and the head and neck 35 questionnaires. So with sample size, this was a Simon 2-stage design. To prove futility, we needed 10 patients in the first round, and then overall 29 patients. This is the schema of the study. So you can see the inclusion criteria. ECOG 0 to 2, recurrent metastatic, 18 to 75 age, adequate organ function, and having financial constraints for nivolumab, cetuximab, or pembrolizumab, and with some measurable disease. And we gave them EMF every 28 days. And this is the time point we did clinical examinations, and then quality of life at baseline and at 3 months, cfDNA and CTC at baseline and at 3 months. And these were the primary endpoints. So we assessed this for eligibility 49 patients, and mind it, this was done in 20 and 21. The The whole world was ravaged by this COVID-19, and we did this study during this period of time. We excluded 10 patients because of ineligibility and enrolled 35. And this results I'm going to present is of 34 patients evaluable at this present time. QL analysis of 30 patients, and for CFDNA and ctDNA, I'll be focusing only on ctDNA. This is for 28 patients only. So this is the regimen. These are the results. So you can see that for these 34 patients, the average age, the median age is 45. These are mostly males. ECOG PS1 is 65% of patients. Bulk of the patients are buccal mucosa and locoregional disease failing treatment, 85%. And 60% of these patients have received previous radiation also. These are the baseline lab parameters. And this is the result. 5 is the median number of cycles they received, range 1 to 12. And this is the primary objective. So out of these 34 patients, 16 had a PR, 8 had a stable disease, and 10 patients progressed at 3 months. So this gives us an overall response rate of 45%. The disease control rate is 68.5%. And we followed them up to 6 months. Of course, the response waned off, and we were left with a disease control rate of 26% only. The progression-free survival median was 5 months, and the overall survival was median of 9 months. These were the toxicities. You can see mostly grade 2 up to grade 2, rash, mucositis, paronychia, and this nausea, vomiting. These were mostly. Grade 3 were not much troublesome. Rash was in only 5 patients, mucositis in 1 patient. So compliance to treatment was around 88%. And whenever these patients progressed, most of them had to be put to the best supportive care. So this was the last line of regimen of most of these patients. And most of them died of tumor bleed and infections. So summary so far, the clinical part is we have with this EMF regimen, the objective response is 45.7%, PFS 5 months, overall survival 9 months, disease control 68.5%. So these are the quality of life data, and we could show that that only pain had a meaningful improvement. Meaningful improvement means more than 10-point change from baseline. So head and neck 35, pain, swallowing, dryness of mouth, and feeling ill, all this improved. But responders versus non-responders was not much of a difference. So if we compare this with other results, they are very compatible with what Dr. Patil showed, the 3-drug metronomic regimen, erlotinib, methotrexate, and celecoxib. It was in second-line patients, and this is second and third-line patients, almost similar response rates, and the median OS is 9 months. If I have the liberty to compare this cross-trial with nivolumab, what does nivolumab give us? It gives overall survival of 7.5 months in platinum refractory settings. So cross-trial comparison is not the norm, but it gives a decent survival. Now, coming to the second objective, that is circulating tumor cells. Our objective or mission was the quantitative comparison of CTCs at baseline versus at 3 months. So 5 ml of EDTA blood sample was taken, and OncoDiscover liquid biopsy technology was used. This is, as you know, EpCAM-based immunomagnetic separation, and we get the results in number of CTCs per 1.5 ml of peripheral blood. This is how the reports came from Dr. Jayant's lab. And what if this is the first one is the CTC per 1.5 ml of blood, and second one is the CTC clusters. These are the 2 reports, 2 values in the same report. So the median CTC count at baseline was 1 per 1.5 ml of blood. The range was 0 to 4, and mean was 0.9. Clusters, we could not find much. At baseline, it was only in 2 patients, and at 3 months, only in 1 patient. So what was the detection rate at baseline? For 35 patients, 60% had a detectable circulating tumor cells at baseline, and it was undetectable in 40%. This is the bar graph showing baseline and 3rd month assessment. So the gray bar is the baseline, and the green bar is the 3rd month. So for the first patient, you can see at baseline, it was 3, and at 3 months, it came to be 0. For the 3rd patient, you can see at baseline, it's 1, and at the 3rd month, it's again 1. So this is the story of all these 34 patients. You can see from this picture, from this diagram, what happened to the responders and the non-responders. The responders means CR, PR, and stable disease. These patients had a significant decrease, that is the red line, decrease in the number of CTCs, and the non-responders had a significant increase. This was statistically significant. Then we asked the question, can baseline CTC predict the PFS? Of course, this is a very small sample size, and we could not have a statistical significant difference. We again asked the question whether baseline CTC is associated with survival, overall survival. Again, this was not statistical significance. The next question was whether CTC clearance at 3 months, does it have some value? Does it give a meaningful predictive factor for PFS or OS? We could not get anything statistically significant, mostly because it was a very small sample size. Next interesting thing is the concordance. What was the concordance with our conventional clinico-radiological assessment? We found that among these patients, 52.9% had a concordant results. The CTCs speak, the results given by CTC correlated very well with the clinico-radiological responses. But there was discordance in 4 patients, it's 11%. And we could not evaluate it for 35% of the patients. So let us show the examples of concordant results. So this patient is a 45-year-old female tobacco chewer. She had left-sided buccal mucosa, unresectable. So she was started on palliative chemotherapy, 3 cycles paclitaxel. She had progression, and this is when she was enrolled in the study. Started on three cycles, started on EMF. Post 3 cycles, this was the change. This was the baseline. At baseline, you can see there were 3 CTCs, and at 3 months of EMF, after EMF, the CTCs are 0. And this is the clinical response. So the response matches. Second patient, another 32-year-old male, right buccal mucosa, post 3 cycles paclitaxel progression, and then enrolled in the study. Post 3 cycles EMF, he had a partial response clinico-radiologically. And again, we see baseline 2, the CTCs come down to 0. For patient 3, this is carcinoma maxillary sinus, very locally advanced. She received CT-RT and then had a progression. Now she was enrolled in the study. Post 3 cycles EMF, and at baseline, at both the times, this patient had 1 CTC per 1.5 ml of blood. So this was stable disease as per CTC, but and this was clinico-radiologically also stable disease. However, the very next month, this patient progressed. Now, let me show you another example of a discordant result. This is a young chap, smoker, lateral border of tongue, surgery, followed by adjuvant CT-RT. Within 6 months, disease came with a bang. Post, he was enrolled. 3 cycles, after 3 cycles, there was clinical benefit. Imaging showed a partial response, but CTC counts did not go down. So at baseline, it was 1. At 3 months, again, it remained 1. And at the very next month, this patient progressed. I'm showing you the CF DNA data of this patient because this showed a rise from 7.4 to 12.3. But this patient progressed. So the persistence in this case, as well as the previous case, we found that these patients progressed very quickly, and they did the very next month of assessment. So to conclude, this study has the strength that it reports a biomarker in recurrent metastatic head and neck squamous cell carcinoma. It's a very difficult scenario. We have a huge unmet need. This is an India-centric cancer. These problems plague our clinics. These patients are full in our clinics, but we did not have a biomarker until recently. So this study is the first report of this recurrent metastatic population. So limitations is that this was a very small sample size, 35 patients. We can only generate some hypothesis on this. It was a single arm. There was no controls. And we had a very low detection rate despite heavy burden of disease. I'll come to that. And because if the patient has a baseline 0, the comparison becomes difficult. Now, the question was that why was the CTC count so low in recurrent metastatic head and neck squamous cell carcinoma? Is it a special situation? Dr. Jain just mentioned there's a study recently published in oral oncology. This is a good journal. 152 treatment naive patients. They're not exposed to treatment. There, the sensitivity, specificity, accuracy, everything is above 94%. And more than 20 lymph nodes suggest node positive. More than 13 suggest advanced disease. That is what Dr. Jayant and Dr. Pankaj also said. Then the cutoff is for true positive is 3.5 cells. If you see Chinese study, these are recent contemporary Chinese study, they also were done in upfront setting, not in the recurrent metastatic setting. And they showed positivity rate of 81%, and their cutoffs were different. It was their median rate were different, 7.5 per 7.5 ml of blood. And this is 2 per 2.3 ml of blood. This is how they have reported. So what was the reason for this low? Because in recurrent metastatic, we expect that the burden is high, but still we had a median of 1 cell. Probably the setting was different. Upfront, it's different factors work, and at relapse, different factors work. And maybe it's a small sample size effect also. 60% of our patients had received radiotherapy and progressed very soon after that, within 6 months. So we also had, tried to analyze whether this has an effect or not, but we could not find that in the post radiotherapy there was some effect. We could not make it have it significant statistically. To me, it appears, it is a hypothesis that these patients may be even on chemotherapy, and on chemotherapy they had progressed, and then we have enrolled in these patients, many of these patients, and many of these were exposed to radiation. So the quality and quantity of these CTCs may be poor in these patients, and therefore the cutoffs may be different. We don't know the answer. So the take-home point is that EMF is a feasible and safe regimen. It demonstrated a favorable response in platinum refractory settings. It improved quality of life. Until now, we did not have any biomarker established in head and neck squamous cell, and response was judged on the clinico-radiologically. So this novel study and this study published by Dr. Jayant, now we have evidence of an emerging biomarker and we need more studies to show that actually these work, and more interventional studies, not only observational like this. So in future, circulating tumor cells will show promise as a liquid biopsy. And I feel that circulating tumor cells threshold or cutoff values needs to be established differently for upfront and for relapse setting. And we have to go beyond just enumeration. This single cell circulating tumor cells characterization will give us insights, and expansion will also give us for function analysis, the multi-omics things that we are talking about. So I like to acknowledge that this was the DM thesis of one of my bright students, Dr. Annie Kanchanba. And I'm grateful to all my colleagues in head and neck multidisciplinary team at AIIMS, Dr. BRA, IRCH AIIMS. I'm grateful to Dr. Jayant for doing all the lab work relating to CTCs. He has done this so generously for us. And I'm also grateful to NATCO for providing us the allogeneics. And finally, I am deeply grateful to all our enrolled patients and their families who participated in this study in, I think, the most pathetic phases of their life. So thank you very much, and open to questions. Yeah.
So congratulations, sir, for the study. Thanks. A lot. So I had one question about the discordant responses. Do you think the time of sampling of the CTC for a patient who is on therapy, meaning was it done just before the chemo injection or just after or in the morning when he took the erlotinib, do you think this kind of temporal variation might have made a difference? I don't know. That's why I'm asking.
Yeah, we have not specifically looked at this issue because we just had to know the day 28, we took the sample. I'm not confident in saying what time we took the samples. So these pre-analytical variables may be playing a part.
Because
I don't know.
I also faced discordance in a couple of patients, and I'm not sure what to make of that. Maybe there was some relation with the temporal time of CTC or something. So that is the reason I'm asking this. I don't know.
Yes, they may be pre-analytical variables also. Sir, please.
Thanks for a good presentation. And my only concern is you all did the CTCs once the recurrence has occurred and the other thing. have we got any, as a Mr. Patil has highlighted, do we have got data when the primary diagnosis has been done, CTC has been done, so that you could have correlated it much better. See, when the tumor markers like CEA came 30 years ago, we all knew that by the time we did not have a good imaging setup at that time, CEA used to rise. Even till today, increasing levels of CEA in spite of PET and everything, we are in a dilemma. Similarly, if you take this at the primary time of diagnosis, and by the time you do the treatment and repeat it, if that would have shown some response to treatment and CTCs, will it have any impact instead of we looking at it after the recurrence has occurred?
Yes, sir. You're talking about longitudinal doing CTCs. That's the correct plan. I think for the studies should be built like that. They should be done like that so as to exactly pinpoint the value and establish this with level 1 evidence that actually these work. So this study was exclusively in the platinum refractory patients. We did not have the baseline of these patients because they were enrolled later on. So the studies should be designed like that, as we are mentioning, sir, because this only will generate level 1 evidence for these tests in different setting, and maybe for treatment changes, maybe in the adjuvant setting, maybe adding extra to the adjuvants. I think future studies should be done like this, sir.
We see in more numbers.
Yeah.
Thank you, Dr. Pramanik.
Thank you. Thank you.
I would like to say here that the CTC is a technology that we have developed from scratch in India and done it quite differently from what has been done in the West. And the cost differential is also pretty high. But we do understand there are a lot of questions to be answered. We are happy to collaborate with Indian oncologists to figure out how to, bring this up more into the system. Thank you so much. I would now like to go to the third part of the session, and I'm sure you're going to find this very interesting because this is something that is completely new to the oncology space. I would like to call on Dr. Arvind Krishnamurthy to come and introduce Dr. Ken Bloom, who's going to give our next keynote.
Good evening, and hello to everybody. It's my pleasure to be here. I think it's really been very engaging, very insightful for me as well, and looking forward for the session 3. The session is titled AI-enabled image analytics for predictive oncology, and we have an interesting keynote on digital pathology from Professor Dr. Ken Bloom. Dr. Ken Bloom is the Chief Medical Officer in the Advanced Pathology and the Genomic Services at the InVicro and the Ambry Genetics at Konica Minolta company. He brings about more than 30 years of experience in the field of pathology, oncology, telemedicine, and bioinformatics. His role in Ambry has been to sort of expand and strengthen and commercialize the full suite of precision medicine. And previously, Professor Bloom was the president of the Oncology and Immunotherapy for the Human Longevity Inc. company, where he and his team led and developed a commercial product for the leading cancer exome product and commercialized a validating of the neoantigens in predicting from the sequencing part. So Dr. Bloom was also instrumental to be, and as a business strategist, that evolved the Clarion and follow up with the acquisition for the GE Healthcare. I think we are sort of very happy to have him on board, and he's going to speak to us on an interesting topic of AI-enabled predictive oncology in transforming cancer patient care, and with the unit case of HRD status prediction. Over to you, Professor Bloom, and looking forward to hearing from you. Thank you.
There we go. Finally unmuted. Good evening, everybody. really fascinating discussion so far. And I think everybody can tell that this is a changing world. The evolution of liquid biopsy is going to change the way that that we interact with patients, and certainly change the way that we handle patient care. I'm going to talk to you this evening really about the evolution of pathology and what I'm calling the emergence of really next generation digital pathology. I was involved in digital pathology in my residency almost 40 years ago now. Had some of the first patents on telepathology, and it's really come of age. And I say evolution of pathology really because the job of the pathologist has always been to classify disease. So the earliest pathologists, as the cartoon on the left sort of shows, really relied on gross examination to classify disease. Then along came microscopy. We got better and better, special stains, resolution improved all the way up through electron microscopy. But as we all know, morphology itself has its limitations. So I remember as a resident when we were presented with an undifferentiated tumor, if the confusion still persisted following internal consultation, electron microscopy, special stains, we'd send the case out for an expert opinion. Immunohistochemistry at the time was really just emerging, and that was really our ability to see specific proteins within a cell. As the relationship between intermediate filament expression and tumor classification crystallized, what became evident was that the experts that we were sending to were really misclassifying these undifferentiated tumors about half the time, meaning that it was about the same thing as guessing. So adding protein expression to morphology allowed us to refine our classifications of tumors and to better identify therapy options for the newly identified subclasses. That was really our job as pathologists. Going backwards from those new technologies allowed us to become better morphologists. We could go back and recognize features that we had never recognized before. And advances in molecular techniques have provided further insight. So, now today, beyond the H&E, we can subclassify on morphology things like TILs, use immunohistochemistry like HER2, do FISH testing, multiplex immunofluorescence, next generation sequencing, et cetera, that all allow us to classify tumors with more and more granularity. And of course, the reason for that is to give us better insight and improve the care of our cancer patients. We're now at a point where we're poised to gather and organize these features and apply machine learning to subclassify tumors and improve response to therapies. And as a pathologist, my job is now transformed from just looking at a microscope to the way that I envision myself, maybe not today, but probably 3 to 5 years from now, really sitting in a cockpit where I've got all the data available to me to best categorize the disease to give you as clinicians really the best insights into your patient's tumor. So the reason we classify disease at all is to identify patients that are going to respond to therapies. And the best example of how this improved in the lives of patients is probably the classifications of lymphomas and leukemias. So 100 years ago, 120 years ago, at the turn of the 20th century, lymphomas and leukemias were only known as diseases of blood, and 5-year survival was essentially 0. Improvements in diagnostics led to refinements in classifications, new therapies, and better survival. The advent of immunohistochemistry and flow cytometry led to further granularity and classification, as did cytogenetics and FISH. Today, with the development of next generation sequencing, there are close to 150 different subtypes of lymphomas and leukemias, and the 5-year survival is now north of 80%. Now, solid tumor classification is a bit more complicated because, as we've already talked about in many of these lectures, tumor heterogeneity, the tumor microenvironment, and the increasing recognition of the patient's germline variants, all of which can influence a tumor's growth and its ability to respond to therapy. The hope, and I'm going to push back why I think this approach might be flawed, is that precision medicine is a reality, and that by establishing the right diagnosis and identifying the Achilles heel of the tumor through biomarker testing, we can successfully eliminate the tumor. Our supply of weapons against the tumor has grown significantly over the 40 years of my career, evolving from chemotherapy to targeted therapies to antibody drug conjugates, immunotherapy, and combinations of all of the above today. But to use these effectively really now requires increasingly complex understanding of the tumor's ecosystem. Because complete biomarker testing is currently necessary to optimize the initial therapy selection, there can be a significant time gap when the diagnosis is made and therapy is started. We've talked about this significantly already, that there's a large cost to this and that it can take weeks to get the results of next generation sequencing back into your hands to make decisions. Morphology, immunohistochemistry, and PCR can be performed usually within a day or two. And there's other things beyond next generation sequencing. We're going to see the evolution of things like multiplex immunofluorescence, all of these things which might help us get a more complete picture of the tumor's makeup, the heterogeneity of the tumor in the microenvironment. Now, if we insist on complete biomarker analysis before we start initial therapy, we might have to wait the weeks before obtaining results. But I think that next generation digital pathology solutions, coupled with the emergence of liquid biopsy, might change our perspective on how to optimally approach cancer therapy. As I see it, there are several issues that we would like to resolve to optimize a patient's treatment. One I've already mentioned is eliminating the time delay before biomarker analysis is complete. The second issue you've heard a lot about already in other lectures is how we can better assess the effectiveness of our therapies through the use of liquid biopsy. Because of the significant heterogeneity found in most tumors, any therapy that a patient is given will cause selective pressure, allowing those clones unaffected by the therapy to proliferate and become dominant. Since our current methods of monitoring therapy effectiveness are not very sensitive, are costly, and frequently involve radiation, it becomes extremely important that we select the most effective, least toxic therapies up front. However, with the advancement of liquid biopsy technology, our ability to detect treatment failure should be significantly enhanced, allowing us to discontinue ineffective therapies much sooner and allow the patients to move on to other, possibly more effective therapies. When this time comes, the war against cancer may become a series of more effective, less toxic battles. In today's talk, I'm going to use the example of PARP inhibitors to highlight some of the complex issues with biomarker testing and how advances in digital pathology and machine learning might improve the process. Olaparib was approved in 2014 as the first-in-class inhibitor of poly-ADP ribose polymerase, or PARP. PARP inhibitors have shown efficacy and acceptable tolerability in patients with several types of solid tumors, particularly those harboring pathogenic mutations in BRCA1 and BRCA2 genes. Treatment leads to cytotoxic accumulation of unrepaired DNA double-strand breaks. The efficacy of PARP inhibitors can be predicted by assessment of the homologous recombination pathway. While not all patients with homologous recombination deficiency will respond to PARP inhibition therapy, most patients who respond to therapy will demonstrate alterations within this pathway. Just going to do a quick review of DNA repair mechanisms for those that might be unfamiliar or have forgotten. There are several broad methods of DNA repair, which include direct reversal, single-strand repair mechanisms, which include base excision repair, nucleotide excision repair, and mismatch repair, and double-stranded repair mechanisms, including homologous recombination, non-homologous end joining, and microhomology-mediated end joining. PARP1 acts as the first responder that detects DNA damage and facilitates the choice of repair pathway. It is essential for the detection of single-stranded breaks, and lack of PARP1 through inhibition leads to an increase in double-stranded breaks. If double-stranded breaks are repaired by the homologous recombination repair pathway, the double-stranded breaks are repaired effectively. But if the breaks are handled through end joining pathways, the genome may acquire permanent changes that we identify as genomic scars. Let's just look briefly at the process of homologous combination repair. Members of the homologous recombination pathway can be categorized into sensors and effectors. Double-stranded breaks are first sensed by the mRNA complex with the aid of BRCA1. The mRNA complex comes in contact with the damaged DNA, removes about 1,000 base pairs from the 5' end of the DNA strand, creating a 3' overhang. This process is known as end resection. Small proteins, known as replication protein A, then cover the single strand overhang, preventing it from being damaged by nucleases or recoiling on itself. BRCA2 and PALB2 co-localize with RAD51 to displace the RPA. RAD51 then aids in the search for and in the process of invading the homologous DNA. This results in a displacement loop called the D loop. The 3' end overhang will then use the homologous DNA template and synthesize new nucleotides. Once this process is complete, the termination process begins and the DNA will be effectively repaired. Now, blocking the function of PARP significantly impairs the ability of a cell to repair single-stranded DNA breaks and thus leads to double-stranded DNA breaks. Assuming normal function of the homologous recombination repair pathway that we just reviewed, the cell should repair the damage effectively. However, if there is functional deficiency in that pathway, the cell will not be able to repair itself, leading to cell death. This makes PARP inhibition a potentially effective therapy when homologous recombination deficiency is present. Functional HR deficiency can result from a variety of different mechanisms, such as germline and/or some sporadic mutations in key homologous pathway repair genes, such as BRCA1, BRCA2, as well as alterations in gene expression. Deficiencies in the pathway will generate permanent changes that are the genomic scars that I talked about before. Now, there are currently 3 assays, 3 basic ways to identify the presence of homologous recombination deficiency. 2 of these ways utilize next generation sequencing. In one case, looking for somatic or germline alterations in key HRD genes. And in the other case, looking for evidence of genomic scar signatures. More recently, functional assays have been developed. Unfortunately, there's not a lot of uniformity between HRD assays offered between different laboratories. Most have focused on germline and somatic alterations in key genes within the pathway, but functional assays and genomic signature assays may be more reflective of actually detecting deficiency. There are a large number of genes in the homologous recombination repair pathway, and of course, they're not all equally important. Key genes like BRCA1, BRCA2, and PALB appear to be effective predictors of efficacy in most tumors, while other members of the family appear to be predictive in some tumors, but not all tumors. Assays for detecting HRD signatures, these genomic scars, include things like large-scale transitions, which are structural chromosomal aberrations that arise due to inappropriate recombination between chromosomal segments. They also include telomeric allelic imbalance, which are aberrant chromosomal end fusions due to inappropriate end joining, which leads to allelic imbalance after breakage during mitosis. Lastly, loss of heterozygosity results from inaccurate repair of sister chromatids during the SG2 phase of the cell cycle and can be assessed by SNP arrays or by next generation sequencing. More recently, functional assays have been developed, though they are less commonly used in clinical practice. These methods include both direct and indirect assessment. Patient-derived tumor organoids can be treated and then evaluated following exposure to PARP inhibitors, or the organoids can be exposed to X-rays, and then the capacity of the homologous recombination repair system can be assessed to identify those that are deficient. all of these techniques, while not perfect predictors of PARP inhibition response, provide some guidance for patient selection. However, not all tests are created equal, and different laboratories assess HRD differently, leading to potential confusion among clinicians. There are many limitations that I've already discussed, including turnaround time, cost, large numbers of samples that are rejected, increased tissue requirements, heterogeneity that's present within tumor, and the lack of concordance between laboratories. So the real question as a pathologist really comes down to, how can the emergence of digital pathology improve the assessment of HRD status, and especially limit the two current truly limiting features, which are namely cost and turnaround time. So can I effectively predict HRD status rapidly at the time of diagnosis and do it in a cost-effective manner? Can we use the results of molecular and functional testing with machine learning to help us better identify the morphologic changes that homologous recombination deficiency causes within tumors? And can we train an algorithm to interpret whole slide images to predict HRD status? The digital pathology ecosystem is an emerging trend in pathology, and while it may have outgrown its infancy, it's definitely still in its childhood. Digital pathology has mostly been focused on workflows, aiding the pathologist in establishing a diagnosis and providing image analysis tools to serve as an aid for quantitative immunohistochemistry, such as assessing PD-L1 status or HER2 expression levels. Non-pathologists are generally more impressed by these tools than practicing pathologists, who generally find the current workflows cumbersome. They're helpful in providing reassurance for junior pathologists, but for more seasoned pathologists, this is largely unnecessary. One helpful factor is overcoming fatigue, which can be a significant problem in high volume laboratories. What I'm talking to you about today is really an algorithm that's been developed that can help determine HRD status from an H&E slide, a task that can't be performed even by expert pathologists. The same H&E slides that are used to establish the diagnosis can be used to give a predictive score of the likelihood that a given tumor demonstrates homologous recombination deficiency. The H&E slide is first scanned on a whole slide imaging system, and the whole slide image is produced. This can be done in most pathology laboratories. Large academic laboratories almost all have whole slide imaging systems today. But even the ones that don't, there are now relatively inexpensive commercial scanners that are readily available from multiple vendors. The scanned image undergoes some quality checks, which is always important. You need to make sure that things are in focus, that staining is proper, etc. Make sure that the image quality is sufficient for analysis, and then the image is broken down into tiles and the predictive algorithm is executed. The resulting output is a heat map with an overall score given then either by the individual heat map or as an average throughout the entire tissue that was examined. The algorithm itself was developed from 120 high-grade ovarian serous carcinoma cases that were sent to my laboratory for next-generation sequencing testing through our HRD test. Histologic features were extracted from the whole slide images, followed by feature mapping. 70% of the cases were used for training, while 30% were used for validation of the AI model. The algorithm was trained to segment the tumor and predict the HRD status based on the NGS results. The overall accuracy was 99.3%. Now, while this is very impressive, there's a couple caveats that we should probably go over. First, the current NGS testing methodology, while good for assessing HRD status, is not necessarily perfect. So there are things that NGS will miss that might be picked up by other functional assays, for example. And of course, the best way to train these algorithms in the future would not be to predict HRD status, but actually to predict response to the PARP inhibitors. So that would be an excellent overall next step. Now, the other thing that was interesting as we looked at these results was that while the sensitivity and the specificity overall were fantastic, as you would expect when there's 99.3% accuracy, was that there was significant heterogeneity even in the HRD prediction within an individual tumor. And this isn't surprising at all, given the amount that we've already talked about the heterogeneity within tumors. And it turned out that the best predictive power for saying whether HRD was present was really to review the heat map and look for foci of HRD positivity rather than the average across the entirety of the slide. So of course, this is just an emerging technology. For me as a pathologist, this is an incredibly exciting area. It says, can we really take the next generation of what pathologists should be doing, which is really rapidly predicting for you as oncologists and surgeons, the best therapy for patients at the same time that we make the diagnosis. And the theory is that through AI and machine learning, we can take our outputs of next generation sequencing, of multiplexed immunofluorescence, of all these new technologies that we're bringing to bear to analyze our tissue pathology and translate those back into things that we could identify now morphologically with the aid of machine learning. And so what that does is it bypasses the cost and it bypasses the time delays of all of the sequencing that we're currently doing on tissue. Now, you've already had many discussions within this conference about, well, what's the best way to go? Should we be looking at tissue or should we really be looking at liquid biopsies? And because of the heterogeneity and because of sampling issues with tissue, we understand that as we look at next generation sequencing, for example, from different areas of the same tumor, that we don't get great agreement within the tumor with deep analysis. So for example, you saw presentations already that when we look at next generation sequencing of blood, ctDNA, that we can see major differences within 6 months. Well, we've seen those same things when we take multiple sections of the same tumor and place those under next generation sequencing with really concordance of really only about 1/3 of the overall genes. We hope that those are the drivers and are giving you the good information on how to treat the patient, but we actually don't know that. Now, what we hope is that we're entering a new area of integrated diagnostics in deep learning with the potential to transform the way that we practice medicine. I've shown an example where we use readily available pathology images to predict complex pathway changes through machine learning. These types of algorithms could be used to provide near real-time predictions for significantly less cost and potentially greater accuracy than our current methods. While my example was from homologous recombination deficiency, it's pretty easy to imagine similar algorithms being developed for either mutational burden, microsatellite instability, or predicting specific somatic mutations like EGFR mutations, et cetera. When coupled with advancements in liquid biopsy, it really sets us up for a future where optimal care could be given almost simultaneously with the patient's diagnosis and then monitored through liquid biopsy to ensure efficacy of the chosen therapy. Importantly, liquid biopsy could also aid in detecting therapy failures much earlier, greatly reducing unnecessary side effects and eliminating the cost of administering ineffective therapies much sooner. Of course, training in this example was performed relative, again, to HRD testing, and further training should always be performed actually to clinical outcome and not necessarily where we are today to the results of deeper diagnostic testing. So I'm going to end there. I recognize our time shortages and other things going on today and be happy to take any questions.
Question.
Hi, Dr. Kenneth. This is Dr. Sujit here. It was an excellent presentation, I must say, and you actually put us through the entire journey of a histomorphologist. And none other than me can vouch all your sentiments. I am a practicing oncopathologist since 20 years. And as you got excited, I am also getting excited on this topic because if we are going to get all this information on an H&E slide, which we are ultimately going to get after 1 month of NGS and stuff like that, then who else than a morphologist would be very happy. I agree with you that, and it's very exciting, a very nice presentation. I had just one query. In AI or ML or in the deep learning process, can you elaborate on which histological parameters do we take into consideration, or
Yes.
yeah, I mean, are there known parameters or they are still evolving or anything like that?
Yeah, so great question. So it's really a fundamental question of machine learning. Do I do it supervised or unsupervised? Do I tell the machine things that I think that I already know and then let it deduce and basically perform better? Or do I go unsupervised? And I think for the analysis that we're doing today, things need to be unsupervised, right? We're really looking to identify things that, and by the way, the machine won't necessarily tell us what features it saw, right? That's going to be up to us to go back through these learnings and say, well, when the machine could predict this, what do I now see as a morphologist that's going to allow me to say that, yes, I believe this is true, or that this might be a defect, for example, just a side effect of tissue sampling. I think what's most important here is the focus of the job of the pathologist, which is to classify disease. And one thing that we haven't talked about in this conference is the disconnect between surgical pathology morphology and molecular pathology. And the assumption that it's easy to put this information together, because as you guys all know, practicing, it's not, right? And you can go to molecular tumor boards, and I don't care what university you pick around the world, there is not consensus about what any of next generation sequencing data means, right? It's complicated. We're still trying to understand it. And the relevance is always not in just the molecular details. It's always in phenotype. It's always in what is the tumor doing, right? Forget about the individual things. We all know that we see individually the same genetic findings, but the patients behave differently. And the question is why? And it's because it's more complex. You've got a person and their host DNA, all of their variants that are unique to them. You've got differences in immune responses, things in the microenvironment that we're just beginning to understand. So what we really want to do, I think, as a pathologist, is really bring this information to the clinicians as rapidly and as tightly as we can, right? Now, I think the change in liquid biopsy that we've seen allows us not to be necessarily as accurate as we had to have been in the past up front, because now we have a method to actually look and say, is our therapy being effective or not? And so you can change out therapies much quicker. Whereas if you think about what we have to do today, you really have your one-time shot to really decide which therapy you're going to give. And then that course of monitoring takes months. I mean, you know, trying to follow people with radiology, taking not only the expense of CAT scans and MRI, it's also the sensitivity, the exposure to radiation, all of the other things that go along with this. So if you can decide up front with fairly good accuracy, and I believe that this is going to be the next generation of digital pathology, is applying these tools, taking our learnings from multiplex immunofluorescence and next generation sequencing data, and applying that back as a tool for the pathologist to say, not only is this a diagnosis, but this is my best guess at what the first response to first therapy choice should be. I think that that day is probably not here today. We're still 3 to 5 years out in that because we're still trying to understand the morphology. I think that that day is coming. And what I would like to see is, instead of what we're doing today, which is doing our predictions of morphology to molecular findings, do our actual machine learning of morphology to clinical outcome. Because I think there's more nuances there than when we take one aspect of a system, just the genomics of a tumor, and we disregard the microenvironment, or we only look at the microenvironment but disregard the tumor, or disregard the genomics of the patient, we get an incomplete picture, and then our learning isn't as complete as we would like it to be. So that, to me, this is an incredibly exciting time for pathologists. I think that we really need to embrace these tools. And for clinicians, what I would really harp on is I think liquid biopsy changes the paradigm, not just in the way that you're currently thinking about it, which is, can I find what that tumor cell is expressing, or can I find the mutation that's being upregulated so I can pick my next targeted therapy? I would look at it more from the other side and say, can I identify therapies that are failing quickly so I can move on to whatever my next choices are? Because as you know, sometimes the choices are really just combination chemotherapy. You've given examples of low-cost options that work as well as $100,000 therapies in the States. So I think having that flexibility of knowing when things are ineffective to change course and understanding that early on is really going to be the new paradigm.
Thank you, Dr. Bloom, and it was a fascinating lecture. And thank you for your time, and I apologize for the delay. And see you next time.
Thank you.
I would like to invite Dr. K.S. Gopinath and Dr. Ramesh Nimagada to the unveiling of our products. Our next product is, I promised you, it's the talk will be as rapid as our product. So I'll invite Dr. Shafi, who needs no introduction. We'll be quick. I understand it's been a long session, but I believe that this is a game-changing product. This is something that is going to be very interesting. We are going to go fast, and if you send us H&E slides, we'll be faster. Dr. Go.
I know we are running, late. I'll take 2 to 3 minutes, quickly go through. Now, Professor Bloom has made my job easy, talking about predicting genome signatures from H&E imaging. He has described the problem, what problem we have, doing HRD testing using genomics technology is very expensive. And he also described we need multiple technologies in order to actually accurately, identify HRD positive patients, and multiple times the QC failure and TAT. These are the 4 issues. And on top of that, I want to add one more thing. What we did a quick study. We took TCGA public genomics data and analyzed genomic signatures based on what mostly you all do, looking BRCA1, BRCA2 testing. What we identified, only 17% of ovarian cancer patients who turn to be BRCA positive and typically are considered as an HRD positive. Then we little bit move beyond that, see how can we go beyond BRCA1, BRCA2, and we know that homologous recombination repair pathway, the genes which are involved, and consider them along with BRCA1, BRCA2, the percentage of patients who turn out to be positive is 26%. So we got additional 9% benefit, patients who could be actually benefited either by PARP inhibitors or platinum therapy. And when we added additional genomic signatures, as Dr. Bloom was talking about, telomeric allelic imbalance, loss of heterozygosity, and large stage transitions. So we took this what we have identified here 17% of patients which are BRCA1, BRCA4 positive could become, 54%, which is, threefold high. So that means if you do only BRCA1, BRCA2 testing to identify if patients are HRD, could respond HRD inhibitors, PARP inhibitors. So you miss actually 2 out of 3. And this gave us more, understanding. And so we trained our algorithm based on public data and validated it. I'm skipping here, with the data where we had NGS available and identified 99.3% accuracy to identify HRD positive patients. So the accuracy what we got is, it's very high, and this test can be done if we have H&E slide available in 5 minutes. So it brings the TAT to hardly a day. So you get to know that patient is HRD positive. So and also in addition to that, we also look for other genomic markers which are important, like tumor mutation burden, and MSI, as well as PDL1 from same H&E slide. So in a nutshell, what this technology is going to give us is that rapid, precise, and cost-effective report within a short period of time. And with this, I know we are running very fast. So this is OncoPredict, HRD, and IO. We can identify PARP inhibitor biomarkers as well as immuno-oncology biomarkers. So if there are any quick questions, I will take it.
Yes.
So you think this will replace HRD by NGS?
I think this is the time that it will be complementary to what we have. At least it can help oncologists to screen before you sending a test for $4,000 test. You could screen who are the patients who could be sent for that expensive test. One, and second thing is that test will take 3 to 4 weeks. So you have to wait for that period of time. And most of the time, what oncologists do is start immunotherapy or start PARP inhibitors. And by the time you get the report, and then you have to change. The report could be other way around. So at least you get that the flexibility. So it could complement what we are doing as of now.
And you think it stimulates you to take up similar study for other molecular markers like MSI or anything of that
Yes,
sort?
so we already have MSI done, and with similar accuracy as well as tumor mutation burden. So we are working towards others as well, gene fusion, EGFR, hotspot mutation, as Professor Bloom said.
I had one question about the OncoPredict HRD. So is it the same AI platform as mentioned by Dr. Bloom, or is it a different AI platform?
It's same that what we have done is we developed this algorithm and then further validated with the data, with Professor Bloom. It's actually the same.
Okay, so the slides which are the patients which are used, is it the US or UK slides, or is it the Indian slides, which were used to train and validate?
So first we took the some public data to develop the proof of concept, then validated it with the slides which are generated in US, particularly which has the results from memory genetics, HRD results from memory genetics, and validated with that.
So I think we'll need to just double check with some more Indian data before we are able to replace the HRD.
Absolutely, that's why we are here, and work with whoever is interested to validate with Indian H&E slides.
This is one point. I don't think we can look at it as a replacement. Rather, we look at it as a screening tool to look at which patients should you send for those kind of tests. Let's look at it that way first. Then we have sufficient data. That's like a large set. Then it might become a test that you could look at, not going ahead.
Definitely, it is a very important product. So I will just give example of Oncotype DX, which is used in breast cancer. So such a costly product, it is easier to use, mathematical algorithms to predict Oncotype DX. So maybe somewhere this product can be used in that way. And with further validation, definitely, I think it is something which should be more useful.
So,
yeah, absolutely, absolutely.
very interesting product and pricing and the rapid tact also. I just wanted to check whether you have any comparative data with the blood-based HRD testing with this product,
We don't have
or do you intend to sort of go enter that space as well?
So we have some genomics-based comparison. That's how we did the validation for this, not based on the blood base as of now, but we want to validate that.
Right.
Any comparative data in the US itself, like African women with breast cancer and other kinds of things which they have seen in the past when the BRCA was developed and other thing. Did you notice any of the changes as he has rightly pointed out the thing? Indian data may be entirely different from what the American you are used to compare it. Even in the United States, when the verification came for including the HER2, there are a lot of differences of opinion was there. So did you have any such kind of things in the US itself when you are trying to compare between the classical American as well as from the Caucasian other populations and looked at it?
So as we know that, cancer is a complex issue and it's an heterogeneous and there is a variation. And I'm sure there definitely will be the variation and we
As a
have to look into
clinician,
that.
you look at the various aspects of it so that because your costing is very attractive, so that how we can utilize it and try to see, because we have a vast volume of patients are too many in our country, how we can take it forward to generate the data and look what one can do it. Will it be how much useful it is?
So we are ready to collaborate with all of you and give you that you see and you feel this experience and see how it is working before you start prescribing for all your patients.
Also, at the same time, I think once you do this, same thing when the patients are treated, you can do again, follow them up and see whether, not only your test is right or not, it will tell also quite a bit.
Yes, sir. Particularly, it will be very helpful for Indian patients, who has to get platinum-based therapy. At least if we identify HRD negative, there is a high possibility those patients will not benefit from the therapy. So therefore, they can be eliminated.
I think very excellent products and very innovative products. Now, probably I feel you have done your bit and I feel it's our responsibility now to play in with you and maybe take these innovations of this country to the scale. So I think there's a responsibility which lies on all of us, how we make this product more popular, how this product we actually put into our practice. So I personally feel my colleagues will take it seriously, the information given today, especially the last one, because this is price what my patients can afford, and probably at least this information help me. So if you can come up with some other panels like this, so if you can come with a lung cancer panel, for example, or a colon cancer panel based on OncoPredict, probably that will serve my purpose and I can really go, I think, nationwide. Thank you very much.
So definitely, sir, in next 6 months, you will have more products like this, including what we had a suggestion about Oncotype DX, and we will be soon having that image-based similar test which could predict who are the breast cancer patients who could benefit from chemotherapy or who need only hormonotherapy. And also we are working on predicting at least those genomic biomarkers which are in NCCN guidelines, so that every patient, every Indian patient could benefit from this technology.
Thank you. Thank you, Gaurav. Thank you so much. Just in case HRD just took your breath away, there was IO and we do MSI, TMB and PDL1 also. So these are the 2 tests that we are having. Now I'd like to come to the closing keynote by Dr. Sendure Mani, one of the finest scientists in the US in the space of cancer metastasis, a student of Bob Weinberg. And for time, I would like to request him to start his closing keynote. Thank you very much, and I apologize for the delay. Dr. Mani, there is some noise from your end. There's some, I can't hear you. Some background, yeah, better now.
Can you hear me now?
Yeah, I can hear you. Thank you.
Okay, sorry about that. Good evening, and I'm glad you're all having your dinner now. I was kind of concerned it's getting late, and I'll try my best to give my presentation in a few minutes. We have been talking about quite a lot of exciting studies about how circulating tumor cells playing an important role in diagnosing patients and helping in the treatment. So I'm just going to add a little complexity, but I'll also see how I can add some solution about how the heterogeneity among CTCs could create problem, at the same time help us to diagnose patients better. I'm a basic scientist. So these are my disclosures. But one of the things which we have been talking about until this point, everyone was touching upon is metastasis. This is data from American Cancer Society, but it was presented in Economist magazine, showing if patients do have localized disease, which could be independent of the disease type, the survival rate is really high if the patient walks into the clinic early in their diagnosis. But if the tumor is metastasized, it doesn't matter which cancer type it is, the survival rate is very low. You oncologists know very well. I don't think you need an explanation for this. You also know that a while ago, Massimo Cristoforelli demonstrated that presence of circulating tumor cells, particularly in this case, he demonstrated in this paper that patients with more than 5 circulating tumor cells had poor clinical outcome compared to patients who had less than 5 CTCs. This is one of the pioneering work he started while he was at MD Anderson. Now, my friends whom you just heard, both Kathy as well as Klaus, they have really brought this field to a next level, plus many others, to show that how CTC, ctDNA, the liquid biopsy could make an impact for cancer patients, which is really commendable. So now what I'm going to ask a question is that, now we know CTCs are key players. They are very good as a diagnostic tool. Are there a difference in quality versus a quantity with respect to CTC? So that's the kind of question which I'm going to address in the next 10 minutes. So I just want to bring this slide. I got interested in this area because I was trying to understand how the biology of metastasis occurs, where if you look at a normal breast tissue, the cells are beautifully organized by attaching with one another by expressing various cell adhesion molecules. And these cells are attached to the basement membrane. But once these cells get transformed, become cancerous, they still continue to express these cell adhesion molecules. That's why most of the time a transformation leads to benign tumor, not necessarily a metastatic tumor, except in some instances. So then I was the first one, plus few others, we demonstrated that in order for these cells to migrate from one place to another, they need to break cell adhesion, cell matrix interaction. It's called epithelial to mesenchymal transition, which is mostly occurs during embryo development and become latent in adults. That promote metastatic process, which we have demonstrated extensively in many papers. So in addition, while I was in Bob Weinberg lab, I asked this question: when a pathologist, when I look at a tumor at a distant site, the tumor tend to look histopathologically similar to the tumor where they come from. And we know this epithelial to mesenchymal transition play a key role in promoting migration of tumor cells from the primary site to the metastatic site. So based on that, I hypothesized that these cells not only become mesenchymal, but also become stem cells. It's a very short video which Bob was talking about. I thought you could just listen.
Some time ago, a postdoc in my lab, his name is Sundar Raimani. I first met him in Bangalore, in India, the son of a rice farmer from a small, poor village, came to my lab, and he was doing work on this in this general area. And he was interested in the question of what happens when an epithelial cell goes through an EMT, that is a mesenchymal cell. For him, the question was, what's the product when an epithelial cell goes through an EMT? And if you listen to everything I said now, until now, and take it seriously, which itself might be a mistake, then you would imagine that an epithelial cell makes a mesenchymal cell, and a mesenchymal cell is, by all standards, a fibroblast. Fibroblasts are all over their body. They're standard hardware, connective tissue cells, which enable many of our tissues to hold themselves together. And in fact, the answer to this question was so obvious that one might consider it inane. But fortunately, he didn't ask my opinion. He didn't ask what I thought of this question, posing this question, which is good, because I often tell people in my lab to run off on their own and not consult me, since my preconceptions may sometimes sterilize their otherwise rather substantial creativity. So he undertook a fingerprint test. Here are connective tissue cells, bona fide fibroblasts, and here are epithelial cells that have been forced through an EMT. So these are the products of the EMT I talked about before, and these are bona fide fibroblasts. And as you can see from this fingerprint test here, they look different. The products of the EMT are not standard off-the-shelf fibroblasts. They look very different by this fingerprint test, whose details needn't occupy us at present. So he concluded the following, that the product of an EMT is not a fibroblast. Of course, that raised the question, what is the product of the EMT? If an epithelial cell makes a mesenchymal cell, what kind of mesenchymal cell is it? And then he went back to his desk drawer and he looked at a microscopic section of a human mammary duct that he had done together with a surgeon with whom we work at the Brigham and Women's Hospital. Here's a cross section of a human mammary duct. And what he saw here in regions of this mammary duct were cells that seemed to have undergone an EMT, but this was a normal mammary duct. This wasn't some kind of tumor. This wasn't a wounded tissue. This was a normal tissue. And perhaps more importantly, these cells were residing in locations where one knew stem cells could be found. So he then made the speculation that the following speculation, a daring and a rather improbable speculation, that the product of the EMT I told you about before is not a mesenchymal cell, it's a stem cell. And this conclusion, this speculation, promptly earned him the derision and ridicule of all of his lab mates. Of course, I only heard about this later, because what if you have somebody in your lab and you're working on two different subjects? You're working on the EMT and you're working on cancer stem cells. And somebody in the lab says, well, they're really the same thing. The EMT actually creates stem cells. If you hear something like that, then almost always you think that this is the product of a fertile imagination, an overly ambitious, opportunistic postdoc who wants to describe an entire plan of all modern science by uniting everything together. And therefore one is skeptical, as were his lab mates, but he soon shut them up.
Sorry, I just thought I would show that slide to show that through EMT, cancer makes cancer stem cells. Now, these cancer stem cells are floating in the circulation. Now, one other thing, one could always look for cells undergone an EMT in a primary tumor, but the problem here is that it's complex. I think, previous speaker and the previous speaker, everybody explained, like looking at these cells undergone an EMT in a primary tumor is looking for a needle in a haystack, which is not possible. But one could catch them easily in a circulating in the blood. So that brings us to the next issue, the heterogeneity. Now, in the blood, you can have a circulating tumor cells entering the blood through various means. For example, in the early on, leaky vasculature, which formed into the hypoxic tumor, could allow the tumor cells to enter into the circulation without them being very harmful. They are just tumor cells. They enter into the blood. And you can also have cells undergoing EMT, anywhere from partial EMT to complete EMT. Now, we know biology point of view, these cells undergone a complete mesenchymal transition, tend to remain dormant in bone marrow, in various organs. On the other hand, these cells with the partial EMT, they have the ability to revert back. So therefore, they are the most dangerous ones than the mesenchymal one or the epithelial tumor cells entering the vasculature through leakiness of the vasculature. So the one we should be worried about, these cells. Now, these cells, if you ask them at a mutational level, they all would be very similar, but at the epigenetic level and the transcriptional level, they are all different. We know that. So in addition, these tumor cells also interacting with various immune cells. That's one of the very important things we need to look into account. So this is a review article we just published highlighting all this, which is open access, so anyone can access it. So how do you address this problem? So we, this guy Ranga Partha, he's at BD, and this is another person from BD, Bob. They placed this instrument called BD Rhapsody in our lab. So what we are doing now is we are taking the circulating tumor cells and sequencing them with an idea that a transcriptomics will tell us a lot more than anything else. Ideally, we would like to combine more parameters into it. So one other thing initially we did was we went on a simple transcription-based, it's a multiplex qPCR, when you do a qPCR for multiple markers. And we saw, for example, SLUG, which is also called SNAIL2, and CLCA2, and there is KLK5. These are all high relative to patients who are healthy donors in these cancer patients. In fact, the patients who had these factors high in the CTC, they did poorly compared to the patients who did not. But again, this is a crude analysis. So now what we are proposing or doing is that, analyzing them at a single cell level, mainly the transcriptomics. Here is the one, 9 patients we are analyzed already, and there are 100 patients which are under analysis at this point. We take these patient sample and perform a single cell RNA-seq using this Rhapsody. And here are the patients, and you can see we sequenced all the cells, including immune cells, and you could see the CTCs are forming a nice cluster. This is UMAP clustering. And then that CTC population, you could see it's in patient 5. The patient 5, if you look at a response, the patient 5 did not respond to treatment. In fact, that's the only patient who died. The patient had a lot of CTC. But there are other patients who did not who responded, did not have much CTCs. So it could serve as a good diagnostic tool. And we are in the process of pulling out markers unique to the CTC and also on immune cells, with which we could, in fact, go at a better way of classifying them. So, someone talked about IBM Watson earlier. Ideally, one of a friend of mine, Ray Kurzrock, she always feels that we would be able to treat and cure cancer if they are diagnosed early and if they go through precision oncology. Every patient should be personalized and treated individually. If we do that, we could definitely cure cancer. So IBM Watson, at MD Anderson, we had, while it is one of the amazing artificial intelligence mission, it does a miracle, but at the end of the day, you need data. Without the data, it's very hard for any AI-based system to predict. And while people are feeding in IBM, there are many other programs which are kind of using these kind of tools to come up with AI-based prediction. What we have done is that, so here is a slide. I found a company called Ilon Precision Oncology, where, if you think about a cancer patients, with the standard treatment, you all know only some patients benefit, but not many. But with the genetic profiling, it could be either CTC or a ctDNA or a tumor DNA. One could classify the tumors into multiple groups, and then you could give them treatment accordingly. So as we kind of talked again earlier, that you can feed in this multi-omics data, into something like a Watson or supercomputer once we have a lot of data gathered, and that will spit out an information about more toxic, more effective, less toxic individualized treatment plan, which an oncologist should simply, should be able to use it. But unfortunately, we are not there. We are getting there. For example, Mr. Morgan also putting together an amazing tool with which one could do exactly this. So what we have done, Dr. Seventhi Limay and myself and a few others, that we have created this company called Ilon Precision Oncology, where the genomic data could be passed on to the brain of these people who are advisors, and who then spit out some information or provide an input which are more effective, less toxic, individualized treatment plan, which has been going well. It's called Ilon Precision Oncology. So we have already partnered with various institutions and hospitals around the world, and we are looking for active participation by clinicians as well as individuals. If anyone of you interested in learning or understanding more about it, please email at partners@ilon.com. And that's the website, ilon.com. And with that, this is the team who is doing the work in my lab. We are always trying our best to see how we could understand the biology. Through biology, we could come up with a better treatment. Thank you all for making a time. And I know it's too late, and looking forward to maybe collaborating and working with you all. And you are welcome to email me to money@mdanderson.org, and this is my Twitter handle. Thank you.
Thank you, Dr. Mani. Actually, we got a 2 for 1. Thank you for bringing in Dr. Weinberg also. Thanks a lot. Any questions for Dr. Mani? Right. Thank you so much, sir. And I apologize for the delay. Thank you.
No problem, no problem. Thank you.
If anybody would like to comment in last comments, I'll be happy to take them. Comments, please. Right. All right. Thank you. Rick, I would like to invite Mr. Rick Kamble, our director, to give the word of thanks.
We really apologize for a little bit delay as the program was extended, but you had been a very, very, let's say, interactive audience. So first of all, let me thank you to all the oncologists who came all the way throughout the country. Some of the physicians, they travel across the pond from all the way Los Angeles to here. So I really appreciate you spending the whole day with us. And I really want to thank you for being part of our journey and also being part of the new Precision 2.0 new era. As the researchers, as the investors, as the promoters, we have done our job. Now, the onus, as one of the physicians mentioned, that the onus is all the clinicians community to help us go to the next step. All the speakers, they did share a great deal of details about the precision oncology and what needs to be done. And amazing clinical inputs. What a fantastic talks today. Thanks to all of the attendees for their in-person and online presence today. Thanks to event organizers, the Mice Ideas, Mr. Rajesh Sharma, for doing a fantastic job today to putting together this event. All the team of One Cell Diagnostic, OncoDiscover, for their tireless efforts for last 10 days to put all this information and the details of this conference together. Again, I know it's a little bit late. So with that, I would like to thank you, all of you again, for attending to this conference. We look for more future conferences of such a kind and a very close collaborations with all of you guys to challenge us to do more research to improve millions of cancer patients and their life. Thank you all. You have a nice evening. Please enjoy.
Thank you very much. The conference is closed and I'll like to invite you all for dinner. Thank you.